So, good morning, everyone. Yeah, am I? So, today we are starting with which one? Marathon classes. And of course, the intention is to help you with the important area of the NCERT and patiently listen the class. Definitely, you will got the impact of this in the coming NEET exam, right? So definitely, so we will discuss about all different topic of the botany from where I am finding it is important for the NEET exam. Definitely, I will not deliver any concept in detail. We will touch the important area from where there is a possibility of asking the questions in the exam. So patiently listen it. And I commit you in the beginning itself, you will got a minimum advantage of 5 questions in NEET. Minimum. So that will going to find you at the which date? 12th. Getting the things or not? So I am just going to cover the all important aspects in these 3, 4, 5 hours. Getting the things. So patiently be with us and listen the things carefully. So it's not like what you listen today and you also make a mistake in which one? 12th. No, we are just trying our best to deliver the things, right? Right. Okay, children, so let's start. Just give me a moment. So we are starting with genetics. Nothing, that technician was there, no, just now. If it possible, send him. Yeah. So we are starting with what? Genetics. Principle of inheritance. So first of all, you keep some formula in mind. Important formula that will help you in the Mendelian genetics. And what are the important formula? Types of gamete equal to 2:n. Types of phenotype also equal to 2:n. Types of genotype equal to 3:n. Types of Punnett square equal to 4:n. Right? So 4:n. So these are the things you keep in your mind. Now here, what is the value of n? N equal to number of different heterozygous. You can, I'm just telling you how to apply the formula by saying type of genotype. Suppose that someone going to ask you capital A, capital B, small v, capital C, small c, crash with capital A, small v, capital A, capital B, small v, capital C, small c. So it is the selfing of a what basically? 3 heterozygous, trihybrid cross. So how many heterozygous is there? 3 heterozygous is there. These formula, mind it, we only apply for F2 generation result when we use the perform the selfing of F1. So here what's happened? Three heterozygous is there. 1, 2, 3. So what we will do? 3 is to the power 3 equal to how much? 27. Answer. So type of genotype be just types of genotype be determined very easily with the help of this particular formula. So these are the formula you can use to determine any things like gamete number, type, So is audible? Is audible now? Yes. I think some battery issue was there. No problem. See the things. So once again, I am just revising this earlier slide. What I told about monohybrid cross, I told Mendel have taken parent always as pure life. In Mendelian genetics, either it is monohybrid, dihybrid, trihybrid cross. You will find parents are always pure life. And phenotypic ratio we report in F2 generation is how much? In F2 generation, phenotypic ratio is 3:1, genotypic ratio is 1:2:1. What I wrote in Mendelian genetics, F1 resemble with either of the parent. Out of two parent, it will resemble with anyone. Getting the things? And to whom it will express, who will express is called dominant. Suppose that out of tall and dwarf, tall is expressing, so tall is the dominant. Type of phenotype, I already told you the formula. Type of genotype, I already told you the formula. Now I am moving ahead. Now I am going to deal about which one? Incomplete dominance. Incomplete dominance. Right. So if we talk about incomplete dominance, these incomplete dominance studied by whom? Correns. Studied by Correns, who work on discovered by which one? Correns. Right. Now what happened? F1 in incomplete dominant not resemble with any of the parent. Any of the parent. And in F2, both genotypic and phenotypic ratio, both genotypic and phenotypic ratio are same. And what is this? That is 1:2:1. Able to understand? So this is in the case of incomplete dominance. F2 resemble with some of the example of incomplete dominant. Flower color in Snapdragon. Another example of incomplete dominant, flower color in 4 o'clock plant. It is called Mirabilis jalapa. Another example is size of starch grain. Size of starch grain. These are the some of the example of incomplete dominant. F1 not resemble with any parent. Mind this line. It may be examiner ask it. F1 not resemble with any parent. F2 phenotypic and genotypic ratio are same. Now we are going to deal about codominant. Codominant. If we talk about codominant, F1 resemble with both parent. Means both express into which one? F1. F2, both genotypic and phenotypic ratio are same. And phenotypic ratio are same. Same. It is in F2 generation. It is the codominant. Here it is what? 1:2:1. Same. F2 in codominance means codominance and incomplete dominance both give same type of F2 generation ratio. That is the 1:2:1. Some of the example you keep in mind. HbA, HbS, heterozygous of sickle cell anemia, AB blood group, Mn blood group, and you can say coat color in cattle. These are some of the example of codominant. That's over. Now we are going to deal about test cross. What is test cross? Why Mendel perform it? It helps to determine the genotype of dominant trait of F2 generation. Test cross is very important, easy also, but important also, of F2 generation, not F1 generation. By default, F1 are heterozygous dominant, so no need to perform test cross. So mind this line, which generation? F2 generation. Able to understand? So Mendel performed test cross to determine the genotype of dominant trait in F2 generation. Both genotypic ratio and phenotypic ratio are same in test cross. And what are this? 1:1. Genotypic ratio also 1:1. Phenotypic ratio also 1:1. And you better to say it is in format of 1:1. In monohybrid test cross, I am just telling you, monohybrid test cross, I am telling you through the example. Capital A, small a, capital small a, small a. Monohybrid test cross, 1:1. Capital A, small a, capital B, small b, small a, small a, small b, small b. Dihybrid test cross, 1:1:1:1. 4 times 1. Trihybrid test cross, capital A, small a, capital B, small b, capital C, small c, small a, small a, small b, small b, small c, small c. How many? 1, 2, 3, 4, 5, 6, 7, 8, 1. 1, 2, 3, 4, 5, 6, 7, 8. 8 times 1. So how many type 1 will come? Depend upon which type of cross is there. Number of 1 equal to number of gamete produced by hybrid. Here 2 gamete, 2, 1. 4 gamete, 4, 1. 8 gamete, 1, 8, 1. So purpose of the test cross is what? To determine the genotype. Test cross is a kind of back cross. Of back cross. So this is the test cross over. Now we are going to talk about, see, suppose that capital W is an unmodified allele. Concept of dominance I am saying. Unmodified allele. So when this capital W undergo in mutation, it forming a new allele, that is the small y allele. These small y allele have 3 possibilities. Either produce same or less functional enzyme, or it produce different enzyme. Different enzyme is called non-functional enzyme. Or it produce no enzyme. These 2 cases, here case 1, case 2, case 3, these 2 cases give different phenotype. Different phenotype. And these small w is called which one? Modified allele. Modified allele, and unmodified allele is dominant, or it is called wild type. These wild type are what basically? Usually dominant. Usually dominant. So unmodified allele are usually dominant and modified allele are usually recessive. Usually recessive. That is over. Now we are moving ahead. Now we are going to deal about ahead. Dihybrid cross. Dihybrid cross of the Mendel, what you know, 2 characters they have taken. So F2 ratio is what? Basically 9:3:3:1 is a phenotypic ratio. And what are the genotypic ratio? Genotypic ratio is 1:2:1, 2:4:2, 1:2:1. Getting the idea? So simply no need to mug up. If you know monohybrid, you can answer everything. Suppose that here monohybrid, I am just taking 1 minute. Monohybrid phenotypic ratio you know, that is 3:1. So you can say the dihybrid phenotypic ratio. Dihybrid equal to simply write monohybrid square. Monohybrid square. Trihybrid you can write. Trihybrid you can write monohybrid Q. Monohybrid Q. Like, so monohybrid square, what happened? 3:1 into 3:1. For multiply it, answer will come. Phenotypic ratio for trihybrid 3:1 into 3:1 into 3:1. Getting the idea? So what the genotypic ratio? You multiply which one? Genotypic ratio, if you want at any hybrid, you multiply monohybrid, 2 times dihybrid will come, 3 times trihybrid will come. So that way we can determine the phenotypic and genotypic ratio. Able to understand or not? I already told you, we can write it like this also: 1, 1, 1, 1, 2, 2, 2, 2, 4. These all are pure line. So how many in dihybrid are pure line? 4/16. How many? These all are monohybrid. How many are monohybrid? 8/16. How many are dihybrid? 4/16. 4/16. So these are the bifurcation of F2 generation I have given. That dihybrid is also over. Now I am telling you some important conclusion. Based on monohybrid, Mendel gives, not exactly Mendel, discoverer of the Mendel, but it is written law of Mendel. Law of Mendel not given by Mendel, keep in your mind. Gives how many law? Through law, or it is called through postulates. One is what? Law of dominance. Because I don't want to confuse you much. Law of dominance, we also not have time. Law of dominance. Mendel himself not given a law. And second is what? Law of segregation of gamete. These two laws explained by which one? Monohybrid cross. Law of independent assortment. Independent assortment explained by dihybrid. Now we are going to deal chromosomal theory of inheritance. Chromosomal theory of inheritance. Who have given it? Sutton and Boveri. And experimental verification given by verification of this inheritance is given by Morgan. Morgan. T.H. Morgan have given the experimental verification. So what these people told, Sutton and Boveri told, they told the inheritance of Mendelian factors and inheritance of gene are of same type. How they are of same type? Both, they told, both chromosome and gene occur in pair. Both chromosome and gene occur in pair. Both occur in pair. Second things what they told, both segregate at the time of gamete formation. At the time of gamete formation. Getting the things in such a way, only one of a pair present in gamete. Present in gametes. Next, what he told? Next, he told chromosome always show independent assortment. chromosome as always show independent assortment. Getting the things? Now, based on the observation of Morgan, that line not told by them. They also told what basically? Based on discovery of linkage. It is not the contribution of Sutton and Boveri, what right now I'm writing. You keep in mind how the inheritance of chromosome and gene will differ. Two pair of chromosome always show independent assortment. And two pair of gene may or may not show. May not show. Means, what are the moral of the story? Moral of the story is two pair of gene may show linkage. Two pair of gene may show linkage, but two pair of chromosome never show linkage. So linkage is the property of gene, not of the chromosome. So in that NCERT diagram, A side, B side, A side is what? A side is gene as well as chromosome. B side can't prove as gene, B side prove only as what? Chromosome. So both A and B side proved as chromosome, and A side only proved as gene, B side not proved as gene. One thing I'm writing linkage. When two genes shown by two genes present on same chromosome and close to each other, shown by two genes present on same chromosome, that is linkage. Getting the things or not? So if we talk about dihybrid test cross used as tools to check either it is linkage or crossing over or independent assortment. It's better to say rather than to say what? Crossing over or independent assortment. How? Listen. If 1:1:1:1 ratio come in test cross, it is independent assortment. I have written in short IA. If 1:1 is coming, it is complete linkage. CL I have written. And if it is anything of 4 type but different from 1:1:1, an example 7:1:7, it is incomplete linkage. So during independent assortment, P is equal to R. Any kind of linkage, P is greater than R. In case of complete linkage, parental is 100%. In case of incomplete linkage, parental is greater than 50% and less than 100%. So these are the sum of the data you keep in your mind. Able to understand or not? So it is over. Now I am going to tell you about mutation. If we talk about, I am talking about gene mutation. Gene mutation, it is also called point mutation. Another name of gene mutation. It is of 2 types. One is frame shift. Frame shift mutation is more dangerous because it changes the entire sequence of amino acid from where it occurs. So due to frame shift mutation, from the place of mutation, entire amino acid sequence gets changed. So it is of 2 types. It is addition or it is deletion. If someone going to ask you who causes more impact, so one is frame shift mutation, another is substitution mutation. Substitution mutation. Getting the idea? So if we talk about substitution mutation, it is also of two types, transition and transversion. So if we talk about transition, what's happened? Purine replaced by purine. Pyrimidine replaced by pyrimidine. And in transversion, purine by pyrimidine. Purine replaced by pyrimidine. And or vice versa. And you better know purine and pyrimidine. Purine is what? Adenine and guanine. Pyrimidine is what? Pyrimidine is what? In DNA, cytosine and thymine, or in RNA, in place of thymine uracil. So when swapping occur between A and G, it is transition. When swapping of nitrogenous base occur between A and C, an example, it is transversion. So getting the idea? So these are the substitution mutation. Getting the things? Examiner will ask, sickle cell anemia is an example of. Can you answer it? It is an example of. Sickle cell anemia is an example of. No one up? Transversion, very good. Transversion. So this is the end of this chapter. Now we are going to move on. Which one? Molecular basis. Molecular basis of inheritance. In molecular basis of inheritance, we are starting about DNA. In DNA, how many type of bond is there? 3 type of bond. 3 type of bond is there. One is what? Ester bond. Between whom? Between sugar and phosphate. Another type of bond is glycosidic bond. This glycosidic bond is present between sugar and nitrogenous base. Third type of bond is hydrogen bond. It is between 2 nitrogenous base. So in DNA, you better know complementary base pairing occur. Adenine pair with thymine, cytosine pair with guanine. Adenine pair with thymine always with double hydrogen bond, and cytosine with guanine with triple hydrogen bond. Triple hydrogen bond is more stable than double hydrogen bond. Getting the idea? So if we talk about DNA structure more, I am just telling you about Chargaff rule. From here question is going to ask. So what Chargaff told, in a double-stranded DNA, these applicable on dsDNA or double-stranded RNA, you can say. Single-stranded Chargaff rule will not apply. So what they told, in a double-stranded DNA, purine is equal to pyrimidine. Purine is what? A plus G. Pyrimidine is what? C plus T. So in a double-stranded DNA, A plus G upon C plus T equal to 1. Getting the things or not? So this is the Chargaff rule. Chargaff rule also proved double-stranded nature of DNA and complementarity base pairing in the DNA. So numericals people can ask from here. They can, if they have given adenine equal to 10%, thymine also become 10%, 10 plus 10, 20%, 100 minus 20 equal to 80%, 80% have two party, 40% cytosine, 40% guanine. So based on that, they can ask the numerical total base pair is that much. So how much? An example I am telling, a DNA have 200 base pair, 200 base pair, adenine 20%. Then how many nucleotide of cytosine is present? So 20%. I am asking nucleotide of cytosine. Because from here direct question coming, so I am telling you, right? So nucleotide of cytosine, how to determine, you better know, I already told you. So nucleotide equal to what's happened? Nucleotide equal to 2 into base pair. So base pair is how much? 2 into base pair, what happened? 400 into 20% adenine, so 30% is what? Cytosine. 30 upon 100, what the calculation will come? This is the answer will come based on the Chargaff rule. Such kind of numericals is going to ask. Now we are going to deal about replication of DNA. Replication of DNA. So during replication of DNA, how the replication occur? DNA replication is semi-conservative, semi-discontinuous, and bidirectional. These are the replication of DNA: semi-conservative, semi-discontinuous, and bidirectional. Right? The strand of DNA form continuously is called leading strand. And better to say, it will take less time to say through a diagram. So I am drawing a DNA, just try to understand. This is a DNA. It is going for replication. So what you are finding during DNA replication, in beginning who require to start the replication in 5' to 3', there is a need of RNA primer. RNA primer. These primer formed by primase enzyme. Primase enzyme. Primase enzyme is a RNA polymerase enzyme. RNA is forming by transcription here. Now main polymerization occur. So here polymerization occur continuously up to the last. Here it is 3', here it is 5', here 5 to 3', always DNA replication occur. So here the DNA will form what you will find discontinuously. Here you will find DNA formed discontinuously. So in complementary to this strand, what you are finding, in complementary to this strand, DNA replication not occur continuous, DNA replication occur in a discontinuous. So this strand is called leading strand, who formed continuously. This strand is called lagging strand. At lagging strand, DNA replication occur in which one? Discontinuous manner. So the DNA of the lagging strand, fragment of DNA first observed by Japanese scientist Okazaki. So it is also called Okazaki fragment. It is also called Okazaki fragment. So complementary strand of leading strand is 3 to 5'. Complementary strand of lagging strand is what? 5 to 3' of the DNA. So if we talk about enzyme associated with replication, so enzyme associated with replication, first you can say about, I'm just telling you, ori site. is the site, it is not enzyme, ori site from where replication start. It is not an enzyme, mind this line. It is a point on DNA, nitrogenous base sequence on DNA. So what is this? Nitrogenous base sequence on DNA. A specific nitrogenous base. A specific nitrogenous base sequence in DNA is called ori site. This ori site is very important for DNA replication to start. If this ori site is not there, what happens? Replication will not occur. Then what you find, which enzyme required? A helicase required. Helicase break H bond. And by breaking H bond, what it do? Unbinding of DNA. Unwinding of DNA done by which one? Helicase. Another enzyme is primase. Primase enzyme perform what basically? Initiate DNA replication. Initiate DNA replication. This is done by primase. After primase, DNA polymerase. Full name is DNA dependent DNA polymerase. Polymerase. So these are main replicating enzyme. Its function is polymerization. After that, I am writing one more enzyme named ligase, join fragment of DNA. So these are the enzyme required during which one? Replication. Another line I am writing, deoxyribonucleoside triphosphate. Serve dual role. It's a line of NCERT. Serve dual role during replication. During replication. Serve dual role during replication. What are the name? Deoxyribonucleoside triphosphate. What dual role it serve? Role number 1, role number 2. Provide energy. Role number 1. Role number 2, provide nucleotide. Getting the things. What happened basically, suppose that I am writing deoxyadenosine triphosphate. Is a what? Deoxyribonucleoside triphosphate. It converts into deoxyadenosine monophosphate. It is what? It is a nucleotide. So it is providing the nucleotide, and when ATP converting into AMP, it is giving energy. Energy as well. Getting the thing. So it serves a dual role. Keep in the mind. Now, an examiner also asked, I think every alternate year, experimental verification of DNA replication. Of which DNA replication? Of semi-conservative DNA replication. Semi-conservative DNA replication given by two scientists. Experimental verification of semi-conservative replication given by two scientists. One is Messelson and Esthal, and another is Taylor. Messelson and Esthal work on E. coli, and he have taken N15 heavy nitrogen. They work on, these are non-radioactive, examiner will ask, non-radioactive. They work on prokaryote, E. coli, and Taylor work on eukaryote. Same year both work, but who worked first? Messelson and Stahl worked first. Then Taylor work on eukaryote, fava bean, Vicia fava, and he have used radioactive thymidine. Radioactive thymidine he have used as an experiment material. So mind it, heavy nitrogen, non-radioactive, and here radioactive thymidine they have used during the experiment. Able to understand? That is used by whom? Taylor. And Michelson and Stoll have used an E. coli, that is a, which one? Heavy nitrogen. Who say DNA replication is semi-conservative. DNA replication is semi-conservative. Told by whom? Watson and Crick. Semi-conservative mode of replication is given by which one? Watson and Crick. So this is about DNA replication. Now I am going to talk about central dogma of protein synthesis. Central dogma of protein synthesis. Getting the idea? If we talk about central dogma of protein synthesis, how flow of information occur during protein synthesis? So flow of information, DNA to RNA, information go in which one? DNA to RNA. And RNA to which one? Protein. So information from DNA to mRNA is a bidirectional information. It may possible mRNA to DNA came. So when DNA to mRNA is forming, it is called transcription. And when DNA to mRNA to DNA is forming, it is called reverse transcription. Getting the things? It is reverse transcription. So reverse transcription is also called teminism. Teminism, and discovered by Temin and Baltimore. Who discovered, who told about, who have given the central dogma protein synthesis? It is given by Crick. Central dogma protein synthesis is given by Crick. Reverse transcription given by which one? Temin and Baltimore. During reverse transcription, who form? Examiner will form. Reverse transcription form cDNA. DNA formed by reverse transcription is cDNA, complementary DNA, and formation of mRNA to protein is called translation. Is called translation. And you better know DNA to DNA formation is called what? Basically replication. Replication. Now we are moving on the transcription. 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 DNA to RNA is forming during transcription. So you keep the things in your mind. This is a DNA. Here it is a nitrogenous base sequence ATCG. Here CGAT. Suppose that. And here I am finding promoter. Here it is terminator. Assume. So here what happened? I am just writing, suppose that 5' to 3', 3' to 5'. Why I am saying this one has 5' because referential is promoter. This one has 3' because referential is promoter. Toward promoter 3' is here, so it is called 3'. Toward promoter 5' is here, so it is called 5'. So during transcription, keep something in mind. Keep something in mind during transcription. What's happened? 3 to 5' act as template. 3 to 5' of DNA act as template. 5' to 3' act as coding strand or sense strand. and 3' to 5' act as template or non-coding strand. Or better to say, coding, non-coding, you know na, template or anti-sense strand. Sense or anti-sense we are using na, anti-sense or anti-sense strand. And what is the very important line you keep in mind? RNA is like any kind of RNA. Either mRNA, tRNA, RNA, doesn't matter. RNA is like coding strand. Like means not 100% copy. You don't say 100% copy. And complementary of template strand. Complementary of template strand. So this is the transcriptions. So here this is the template strand. So here it is 3 to 5'. Always transcription occur in promoter to terminator direction. So transcription direction will be what? Direction of transcription will be this. It will from 5' to which one? 3. It will from which one? 5' to 3', and this is the direction. So what happened? It will T here, it will come A pair with U, G pair with C, and C pair with G. You please see, this is which strand? Template. This is which strand? Coding. So what you find, children? You find na mRNA or rRNA or tRNA, it is which one? RNA strand. It is complementary to coding, or it is like, it is complementary to template, or it is like coding. Only difference is what? In thymine place uracil came, because thymine is not, it is not present in RNA. In place of thymine, uracil is present. Getting the things or not? Right. So this is about which one? This is about transcription. Another things about transcription I am telling. Steps of transcription. Steps of transcription. If I talk about steps of transcription, what you are finding? 3 steps is there. Initiation. Second step is elongation. And third step is termination. Termination. Getting the idea? Initiation, how it occurs? When along with RNA polymerase, who bind? Sigma factor. When RNA polymerase along with sigma factor bind at promoter of the RNA, what happened? Initiation of transcription occur. After initiating the transcription, who detach? Sigma factor detach, and what happened? Alone RNA polymerase or form elongation. And when with RNA polymerase, who bind? Rho factor bind. Termination occur. So that is the reason sigma factor is called initiation factor, and rho factor is called termination factor. Termination factor. Getting the idea? So this is about the prokaryotic transcriptions over. Now I am going to tell, before eukaryotic transcription, I want to say one more thing. Properties of genetic material. Properties of genetic material. If we talk about properties of genetic material, replication. Who have capacity of replication, that become a genetic material. Second property, stable. Stability is the second property. Stably transfer the character in the next generation. Another property, chance of slow, scope of slow change. Scope of slow change. By which process? By mutation. by mutation. And what are these? Express like Mendelian factor. These four are the property of a genetic material. So out of DNA and RNA, who is the better material? DNA is a better genetic material. Genetic material than RNA. Why? Because it is more stable. Why DNA is more stable? Many reason is there. In DNA, deoxyribose sugar is there. That is less reactive. Ribose sugar is more reactive. That is present in RNA. In DNA, thymine is there in place of uracil. That also provide DNA more stable. Able to understand? Both DNA and RNA can replicate. So it done by do. It is better with DNA. A scope of fast changes in the RNA. DNA go for a slow changes. Mutation is more in RNA. So here RNA is not suitable. And RNA express better as Mendelian factor. But because of stability is more, so DNA in evolutionary process will become a major genetic material. Otherwise, you try to understand, world is RNA world. In this planet, RNA came first, not DNA came first. Because of stability, later on DNA become more stable. Getting the thing, and it act as a better genetic material than whom? RNA. Now we are moving ahead. So now we are going to deal about eukaryotic transcription. Eukaryotic transcription. You keep one thing in mind. In eukaryote, a split gene is there. A split gene means what? Information of a gene is not present together. It is splitted in different parts. This is the functional information. This is non-functional information. This is a non-functional information. What you are finding here. So when it is going for transcription, a RNA is producing. That is called hnRNA in eukaryote. hnRNA produced in which one? Eukaryote. Able to understand or not? hnRNA is producing. These hnRNA undergoes in a processing. During processing, what's happened? During processing, three events occur: capping, telling, splicing. So capping at which one? 5 prime. And this cap is made up of 7-methylguanosine. Telling at 3 prime. And it is made up of poly-A, repeating unit of adenine, polyadenine. It formed in a template independent manner. Formed in template independent manner. Formed in template independent manner. Able to understand? This telling occur at which one? 3 prime. And splicing is removal of non-coding part. When that part formed by the intron, removal of that is called splicing. And after that, what happened? The mRNA form have 5 prime cap of 7 mg. Here it is exon only, and here it is a tail of which one? Poly-A, and it is called mRNA. So hnRNA undergoes in this process and then convert into a mature mRNA. Exon is what? Coding part. Coding part of DNA. Intron is non-coding part. Non-coding part of the DNA. Getting the things. The information of exon present in protein, intron information not present in protein. Who is present in intron? Intron are that gene in our body who not expressed today, who expressed earlier in our ancestor, and that is the reason NCERT wrote a line, presence of introns is a remanence of NTQT. Presence of intron is a remanence of NTQT. Getting the things or not? Intron is our ancestral character. Getting the things? Ancestral gene, who is not a functional gene nowadays. Genetic code. Genetic code. Getting the thing. So if we talk about genetic code, I am writing character. Character. Triplet. Second character, commaless. Third character, it is nearly universal. Fourth character, it is non-ambiguous. Fifth character, it is degenerate. So triplet means 3 nitrogenous forming a code. An example, UUU. Every 3 nitrogenous base code amino acid. Commaless means between 2 nitro triplet, who is not there? No sign of punctuation is there. It is a wrong way. No sign of punctuation is there. Nearly universal means one code responsible for same amino acid. If we not talk about few exceptions, same amino acid in every species. And due to this property of the genetic code, who is possible? Genetic engineering is possible. Genetic engineering is possible due to these nearly universal nature of genetic code. You are adding a bacteria gene in human gene in bacteria, and they are producing same protein because of universal nature of the genetic code. Every word have the same meaning in all country. You think like every word have the same meaning in all country is universality. Non-ambiguous. One code is responsible for only one amino acid. Its meaning is what? One bird have only one meaning. A bird have same meaning every country, universality. One bird have same meaning is non-ambiguity, and there is many word for the same meaning is degeneracy. More than one code for one amino acid. And due to this property, degeneracy, we can't determine mRNA from amino acid. If I know amino acid sequence, we can't say mRNA sequence. Because of which property of the genetic code? Degeneracy of the genetic code. It is not possible to decode mRNA from which one? Amino acid. Getting the idea, understanding the idea, any doubt about, right? Okay, and of course, another property I left one, non-overlapping. Non-overlapping means AUG, CUCG, it is like this. In a one nitrogenous base become a member of only one code, not of another. It not happen like it is correct. If I am doing like this, it is incorrect. Getting the things or not? So it is what basically non-overlapping. Able to understand? Now we are moving ahead. Now we are going to deal about protein synthesis. Protein synthesis. If we talk about protein synthesis, for steps simply I am telling. So first step is activation of amino acid. Require ATP. And during activation of amino acid, enzyme is peptidyl transferase. No, sorry. Amino acyl tRNA synthetase. Enzyme is amino acyl tRNA synthetase. Second step is charging of tRNA. Charging of tRNA. During charging of tRNA, what's happened? Amino acyl tRNA complex bind with tRNA, and it is going to form such kind of tRNA. This is a tRNA, and at 3 prime, who will attach? Amino acid will attach. It is 5 prime, 3 prime. It is a D arm. D arm recognize amino acid. It is anticodon arm. It is T psi C arm, and it is 5 prime. 5 prime always start with G. 3 prime always terminate with CCA. CCA, 5 prime always start with G. Here 3 prime, who is attaching? Amino acid attaching, it is charging of tRNA. So it is the second state of the translation. So up to charging 1 ATP required. Up to charging 1 ATP required. Now we are going to deal about which one? Translation. Translation. Translation, if you talk about, 3 steps are there. Initiation, elongation, and termination. Termination. Initiation of the translation, what happened? During initiation of the translation, with first codon, who is attaching? tRNA is attaching. Here who require? GTP require. Here it is P site, here it is A site. P site is peptidyl site, A site is amino acyl site. So here what happened? That event occurred during initiation. During elongation, what you will find? During elongation, at second codon, what happened? Who will attach? Amino acid will charge, tRNA will attach. First codon, second codon, getting the things or not? As both will attach, it is detached from here and it is joined by which bond? Peptide bond. So peptide bond formed between 2 amino acid. Who form it? 23S ribosomal RNA in prokaryote. In prokaryote. And in eukaryote, 28S ribosomal RNA in which one? Eukaryote produce the peptide bond. For peptide bond formation, energy required but not ATP. But not ATP. Able to understand? For transport of which one? Charge tRNA at the site of protein synthesis, who require? GTP require every time. Able to understand? So now what happened? Translocation happened. So for translocation, what you are finding? During translocation, ribosome shift one nitrogenous base. It requires GTP and enzyme translocase. Enzyme translocase in the translocation. Getting the idea? Now we are going to talk about, it is elongation, then termination. Termination occur when who come? When UAG, UAA, UGA come at which one? mRNA. A stop codon. These are what basically stop codon. Then what happened? They came on mRNA. Then termination occur. Termination require GTP and termination factor. Termination factor. So during protein synthesis, you keep in your mind during protein synthesis, for every amino acid, 1 ATP and 2 GTP required. For every amino acid, 1 ATP and 2 GTP required during which process? Translation process. So translation is you can say during protein synthesis process. During which process? Protein synthesis. So this enzyme I told in the earlier slide, these peptide bond formed by an enzyme. What is the name of enzyme? Peptidyl. Tell me, what is the name of enzyme in translation? Peptide bond formation. Peptidyl transferase. It is a ribozyme. It is a ribozyme. It is a ribozyme. It is RNA. Here a student asking, sir, here which energy? Energy which released during breaking of which one? Amino acid from tRNA used in the formation of peptide bond, not ATP. Energy when released, when amino acid detached from tRNA, that energy used in the peptide bond formation. Now we are going to talk about regulation of gene expression. A regulation of gene expression. So if we talk about regulation of gene expression in prokaryote occur at transcription level. Occur at transcription level only. Getting the idea? In eukaryote, regulation occur at first transcription level. Second, during transfer of mRNA from nucleus to cytoplasm. Third level is during processing of hnRNA into mRNA. And fourth level is translation level. So there is more way of regulation of a eukaryotic gene, but there is only one way of regulation of a prokaryotic gene. So we are going to deal about regulation of gene expression in prokaryote. So of course we are going to deal about regulation at transcription level. So I am going to tell you about lac operon. Lac operon. 2 minutes, give your attention. This is what? This is a promoter for regulator gene. Promoter for which one? Regulator gene. This is regulator gene represented by I. It is acting as an inhibitor. And here it is promoter for a structural gene, operator, and here it is what? A structural gene. A structural gene. The structural gene is ZYA. Now what happens, children? Keep in your mind. These regulator gene you keep in a mind is a constitutive gene or housekeeping gene. Or housekeeping gene. They work all the time, 24 hours. So how they work 24 hours? Here RNA polymerase bind. Here this is going to form the mRNA. And mRNA undergoes in translation and forming a protein. This is a active repressor protein. Active repressor protein. These active repressor protein, what to do? Bind with the operator site. Where they bind? Operator site. Many times I told promoter and operator have some overlapping area. So when operator is going to block by active repressor protein, part of promoter also not become visible, and due to that reason, what happened? The enzyme RNA polymerase for this structural gene not able to bind at which one? These promoter site, and who remains switch off? A structural gene remains switch off. So what happened? No transcription, and no translation occur. No translation occur. So what you are finding here, normal situation is structural gene is switch off, and who did it? Regulator gene did it. How it did it? By releasing a active repressor protein. What this protein do? They block the operator. Part of promoter also get a structural gene. Promoter also get blocked, and RNA polymerase not able to bind, transcription not occur. So normal situation is these structural gene is in switch off mode. So which kind of regulation between a regulator gene and a structural gene? Negative feedback. It acts as an inhibitor, and that is the reason I am saying here it is a I gene. Now what happens? If in the environment of the cell, who will come? Lactose will come. It is a disaccharide. These lactose is a beta galactose side. So these lactose interact with active repressor protein, make it inactive. So as long as in the bacterial cell environment, who is present? Lactose is present. These active repressor protein remain inactive. It can't block the operator. Then what happen in that situation? Here these polymerase will bind at promoter, transcription occur, and after transcription, translation also occur, and they are producing three type of protein: Z producing beta galactosidase, Y producing permease enzyme, and A producing transacetylase enzyme. These all three enzymes are associated with lactose metabolism, and these enzymes only require when cell have lactose. So that is the reason, see the beauty of the nature. These structural gene, which is a luxury gene, who not work all the time, work only when in cell lactose is there. Because all the genes protein involved in lactose metabolism. Beta galactosidase is a lactose digesting enzyme. Permease responsible for absorption of lactose, getting the things or not. So they are related to what? Basically lactose only. So as long as the lactose present in the cell, it is on, switch on. So lactose acts as an inducer of a structural gene, and that is the reason lactose operon is called inducible operon. Relationship between lactose and active repressor protein is negative. Relationship between active repressor protein and a structural gene is negative. Getting the things or not? Relationship between a lactose and a structural gene is positive, right? So this is about the lac operon, and it is the end of the second chapter. Now I am going to deal about principles of biotechnology, or both chapter of biotech we will deal first. First we are finishing the second year topic, so biotech we are going to deal, right? So if we talk about the biotechnology, first you keep in mind what is a recognition site. What is a recognition site? Recognition site are palindromic sequence. Palindromic sequence on the DNA is called recognition site. Getting the things. Another things you have to keep in mind, what is rDNA. rDNA is also called chimeric DNA. So rDNA have what? Suppose that here it is a plasmid, and along with this plasmid there is a desired gene of human being is there. It is a plasmid and it is a human gene. So alien gene is there, na? Human gene is a alien gene here. So this DNA is called which one? rDNA. Recombinant DNA. Able to understand or not? Another things we are going to deal about, which one? Restriction endonuclease. So what is this? Enzyme released by bacteria for its defense. And these restriction endonuclease used by us as molecular seizure. Bacteria not form for us. Bacteria form it for their own defense, but we are using it as what? Molecular seizure to cut the DNA of our choice. So these restriction endonuclease cut DNA only at palindromic site. It can't cut DNA at anywhere. It cut the DNA only at palindromic site. So how it cut the DNA, you try to understand. Suppose that this is a DNA, GAA-TTC, GAA-TTC. These are the sequence, palindromic, 5' to 3', reading frame is same. Here it is 3' to 5'. This is recognized by an enzyme. The name of enzyme is EcoR1. So EcoR1 is a restriction enzyme who cut the DNA between G and A. That kind of cut is called a staggered cut. And after cut, the end produced here is a stick end. So what the end will produce? G, C, T, T, A, A. After cutting, it will give like this. Another will come what? A, T, T, C, G. Getting the idea? So these are the stick end. So these stick end help during joining the another DNA during rDNA technology. So these are the way enzyme cut. How to give the name of a particular enzyme? EcoR1. So E stand for genus of bacteria, Escherichia. Two continuous letter after first in a small letter is saying about a species of bacteria. Coli, R is what basically? A strain of bacteria, that is RY13, and first is order of enzyme from a particular bacteria. So it is the first enzyme obtained from Escherichia coli RY13 stem. So in such a way, we give the which one name of the bacteria, and it is going to cut in a such a manner. Able to understand? Now we are going to talk about how many type of enzyme bacteria releasing? Two type. One enzyme is protecting the bacterial DNA from RE2, and another RE2 cutting the viral DNA during any infection of the bacteria by a virus. Getting the idea? Now we are moving ahead. Now we are going to deal about which one? Vector DNA. Vector DNA. So what are the character of a vector DNA? During vector role is what? It is responsible for transport of desired gene into host cell. Why there is a need of a vector? Why we not directly transport the desired gene into host cell? Because ORI site is not present on the desired gene. So what we are finding, children, try to understand. If we talk about this vector DNA, so in this vector DNA, you will find following character are there. First character, it must have what? ORI site. Second character, what you will find, it must have a recognition site. Recognition site means that palindromic site, because without this, it will not cut, getting cut by the enzyme, restriction enzyme. Third, it must have selectable marker. Selectable marker. Selectable marker. These three properties must be present in a vector DNA. Able to understand or not? And one vector DNA, it is given in NCERT. Which one? PBR 322. So if we talk about these PBR 322, see in these all three are there. This is ORI site. This is ORI site. Getting the things or not? And you will find here who is there. These are selectable marker. It is ampicillin resistance gene and it is tetracycline resistance gene. It is ampicillin resistance gene and it is tetracycline resistance gene. Now here who is there? Recognition site is there. Usually it is 6 base pair palindromic. So these are some of the recognition site I am telling. These are the recognition site is there. Here enzyme going to come and attack. So this is a recognition site for which one? BAMH1. NCERT give all explanation based on that only. So this is a which one? A recognition site. It is a what? A recognition site. This is what? This is what it is, selectable marker gene. It is also which one? Selectable marker gene. Getting the idea? So what you are finding, all the property is present in this PBR 322 or not, of course. So now what happened when we use BAM-H1 enzyme to cut both the desired gene and vector DNA? Mind this line, we use same enzyme to cut both the desired and the vector DNA. Getting the things or not? I will send the slide, no problem, don't worry, I will send the slide. So carefully listen the things mainly and getting the idea. So what I told, when we use BAM-H1 enzyme to cut both the desired gene and which one, vector DNA, what you will find, BAM-H1 will cut here. It will cut here. BAM-H1 will cut here. So when it will cut here, there are two possibilities. When we use the DNA ligase, two possibilities there. Either here self-ligation occur or new gene will add. So here if new gene will add, what you will find? The old gene become inactive or not. In the yellow segment gene, blue segment part it came. So what happened? You keep one thing in mind that is very important. Insertional inactivation. What is this insertional inactivation? When we add a new gene between old gene become inactive. Old gene become inactive. So you please try to understand, when we are attaching the new gene at BAM-H1 site, who will become inactive? Tetracycline resistivity become gone now. So what we are finding, when we are using BAM, can I write a line? Can I write a line? When we use BAM-H1 enzyme, tetracycline resistance gene will there or not there? Not there. So in a recombinant, tetracycline resistivity is not there. Tetracycline resistivity is not there. When not added, in a non-recombinant, tetracycline resistivity occur. So can we say in recombinant resistivity for which one? Tetracycline not there, and in non-recombinant tetracycline resistivity is there. So can we say tetracycline resistance gene acts as a selectable marker to differentiate between recombinant and non-recombinant. Can we say this line or not say? Yes, na? Can we say na? Ki tetracycline resistance gene act as a selectable marker to differentiate between recombinant and non-recombinant. Now you can ask a question, sir, why you are using why in PBR 322 there are two selectable markers are there. So you try to understand a problem. You try to understand, you got the solution of the problem. See, here is a culture medium. In a culture medium, many bacteria is there. Millions of bacteria is there. And in this culture medium, I am adding a plasmid. It is a plasmid. But we are in a doubt that either it is recombinant or non-recombinant. Because we simply use the enzyme to put the DNA and use the enzyme to join. There are two possibilities there. If foreign gene will join with the vector DNA, what happen? Recombinant will form. If self-ligation will occur, non-recombinant will form. So we are in a doubt of mind. You cannot say after looking a plasmid either new gene is added or not added. So we are in a doubt of mind. We adding the plasmid, or it is a vector DNA. Either it is recombinant or not recombinant. We are in a big question mark. We don't know. There are two possibilities. It may be the plasmid we are adding is recombinant or non-recombinant. Recombinant when foreign gene added. Non-recombinant when foreign gene not added. Now, bhaiya, I am just introducing this desired gene into vector DNA. Sorry, vector DNA with desired gene, so-called desired gene, either we don't know, into the host cell. These are the host cell. Now, it will enter in any one, one will enter in one cell. It is millions of bacteria. It will enter in any one of the bacteria. Now I am asking you, you tell me where it have entered. Where it have entered. So you can't say by saying, sir, in this bacteria, you can look on that, in this bacteria it enters, sir. You can't say it. So how to deal it? I am telling you. So can I say in culture medium, two type of bacteria is there. In culture medium, two type of bacteria is there. One is what? One is what? Non-transformant. One is non-transformant. And another is transformant. You can say, sir, what is this non-transformant? Non-transformant are, suppose that bacteria enter in this cell. So this is transformant. These plasmid, not bacteria, plasmid enter in this cell. So this is transformant. The rest all are non-transformant. Here I wrote, to enter here, how to know? So transformant you got then or not? Who got the plasmid from outside is transformant. Who not got is non-transformant. NCERT wrote a line or not? Normal E. coli have no resistivity against any antibiotic. Any antibiotic. So what happened? When we add here ampicillin, who will die? All non-transformant will die. Who will not die? Transformant will not die. You can ask her why transformant will not die. See the things. Few slides back. Here, this is the plasmid and this is ampicillin resistance gene. In every plasmid we added there resistivity of ampicillin resistance is there or not. So I am telling you recombinant or non-recombinant. Both have ampicillin resistivity, agree or not? Tell me. Ampicillin resistivity, we not change anything here, So both recombinant and non-recombinant is a part of transformant. So in transformant, resistivity for which gene is there? Ampicillin resistivity there. So who will not die? The transformant will not die except transformant. All non-transformant will die. So here ampicillin resistance gene help us to differentiate between transformant and non-transformant, and tetracycline resistance gene will help us to differentiate between recombinant and non-recombinant. How? So out of many bacteria, only one bacteria will survive. So we remove the living bacteria, allow them to grow. After some time, it becomes two bacteria. We will take two replica plate. I paste one here, another here. I am adding here tetracycline. If tetracycline kill them, means it is recombinant. So another copy of recombinant is with us. We will use it. So this is about that concept. Now I am going to tell you about blue-white screening. Blue-white screening. Getting the idea? So in blue-white screening, what to keep children in mind, try to understand. In non-recombinant and recombinant. In non-recombinant, who produce? Beta galactosidase produce. In recombinant, no beta galactosidase. No beta galactosidase. So when blue beta galactosidase is there, it reacts with which substances? Chromogenic substances, and it give blue color. So when we add chromogenic substances, non-recombinant give blue color colony because its gene is not inactive, its gene is active. In a recombinant, what's happened? When we add the desired gene, that desired gene attach between beta galactosidase gene. So when the new gene will attach, old gene become inactive. Getting the things or not? Old gene become inactive, and when old gene become inactive, due to insertional inactivation, no beta galactosidase. Why no beta galactosidase? Due to insertional inactivation of X-gal gene that produce beta galactosidase. Getting the idea or not? Now what you are finding, children, here if chromogenic substances, if we add, it will not give blue colony because no beta galactosidase is there, no reaction will occur, so no color will come, so no color give white colony. So this white colony showing recombinant, blue color showing non-recombinant. So by color change we can say who is recombinant, who is non-recombinant easily in a blue-white screening. It based on the principle of which one? Insertional inactivation. Getting the idea or not? So that is also over. Now I am telling about Agro Bacterium tumefaciens. Agro Bacterium tumefaciens is a soil bacteria. These are also called natural genetic engineer. These Agro Bacterium tumefaciens have TI plasmid. Full name is tumor inducing plasmid. Tumor inducing plasmid. Getting the things? So these TI plasmid, this is TI plasmid. In this TI plasmid, there is a DNA who is called tDNA. So it is oocyte, and it is tDNA. So this tDNA is tumor causing. So when these agrobacterium, what happened? Infect broadleaf dicot plant. Getting the things? And cause what basically? Crown gall tumor. So when a agrobacterium infect a normal plant, what happened? It developed tumor in the plant. And tumorous cell release a protein which is enjoyed by bacteria. So it converts a normal plant cell into a tumorous cell with the help of tDNA. And what happening? These tDNA converting a normal cell into tumorous cell, and what happened? Red is not visible. Okay, it convert into tumorous cell, and what happening? Plant release a chemical which required by the bacteria. So how beautiful. A bacteria infected a plant. Listen the steps carefully. Convert a normal plant cell into a tumorous cell. When plant cell become tumorous, these tumorous plant cells releasing a chemical, and that chemical is utilized by bacteria. Means it enter in what? Plant, and force the plant to release the choice of chemical of bacteria. And that is the reason I am telling it act as a natural genetic engineer. So we learned the biotechnology from these bacteria. If bacteria can do it, we also can do it. Nowadays, TI plasmid use extensively as vector. Why? You can say, sir, how vector? So we remove the tDNA part, we disarmed it, we use it as a disarmed plasmid. We remove the tDNA, and in place of tDNA, we add the DNA of our interest. Now I am telling you electrophoresis. It is a technique of separation of DNA fragment due to its differential size. Differential fragment length. So see what happened during gel electrophoresis. This is a gel plate where we are adding agar. It is a seaweed. So this is a very good hydrocolloid or phycocolloid. After absorbing the water, it gives sieving effect. So here it is. What basically sample loading well. In this sample loading well, what I did, I added the DNA. This is the DNA. After adding the DNA, I added RE, restriction into nuclease enzyme. Here it is negative charge. Here it is positive charge. Here it is which one? Cathode. Here it is which one? Anode. So when we flow the current, what happened? First, RE will digest the DNA into differential fragment because recognition site is not present on equidistant. So what happened when RE restriction enzyme digests the DNA, different fragment length will create. When we flow the charge, what happening? DNA negative side, negative repulsion, negative positive attraction occur. So DNA will move toward which one? Positive charge. So who is the smallest fragment? Move farthest. And who is the larger fragment? Move slowest. So what I am finding, DNA arranged according to its fragment length during which process? Gel electrophoresis. Able to understand or not? Movement occur due to charge. If negative repulsion not occur, it not move. So movement occur due to charge. Getting the things or not? And differential movement due to size. Is it or not? So who is the smallest face little resistance during the movement? But problem is these things is not visible in the gel plate. So what we do basically, last year a lot of question came from here in the NEET exam. So we just stain it with which one? Ethidium bromide. Ethidium bromide, we stain it, and after staining it give which color? Last year NEET question. After ethidium bromide staining, it give which color? You tell me. It give orange color. And we visualize it which one? UV rays. Getting the things. And after visualization by UV rays, we remove desired fragment by which method? By elution. So elution is a method of removal of DNA from gel. Able to understand? We remove it by which one? Elution. Getting the things or not? So this is about gel electrophoresis. Getting the idea? Now I am going to tell you about PCR. What is PCR? PCR stands for polymerase chain reaction. Polymerase chain reaction. So if we talk about polymerase chain reaction, what you will find method of method of gene amplification in vitro. In a laboratory condition, in vitro, it is a method of gene amplification, PCR. During PCR, 3 steps are there. 3 steps are there. First is what? Denaturation. Second is what? Annealing. And third is what? Polymerization or extension. So suppose that this is a DNA. At high temperature, we perform it denaturation. Both strands will denature. It is greater than 90 degrees Celsius temperature required for that. So it is the first step, denaturation at high temperature. Hydrogen bond get broken. Now we use DNA primer, mind it, not RNA primer. We use which primer? DNA primer. So we are adding DNA primer, 5' to 3', 5' to 3'. Getting the things. This is the DNA primer. We attach it during which process? Annealing. And we do it around at 56 degrees Celsius. Then we take an enzyme, Taq polymerase. And Taq polymerase perform which one? Extension. So Taq polymerase enzyme we take from Thermus aquaticus bacteria, which is stable at high temperature. Getting the things or not? It is done by Taq polymerase. It is what? Basically extension. And getting the things or not, it obtained from Thermus aquaticus bacteria, and that is the reason its name is Taq polymerase. Are you getting the idea? So these are the 3 steps of the PCR. Denaturation means breaking of H bond, annealing means attachment of RNA primer, extension means polymerization. Polymerization, annealing, addition of DNA primer. Here primer is not RNA, DNA primer. And here denaturation, breaking of H bond. Breaking of H bond. So this is about which one? Polymerase chain reaction. PCR. Getting the idea? Now we are moving ahead. Now we are moving ahead. Downstream processing. One line I am saying. Downstream processing. Everything start from isolation of product. Isolation of product to everything. Isolation of product and before marketing. Before marketing is called downstream processing. Is called downstream processing. Getting the idea? So isolation of product, it include, if you will say, isolation, purification, testing, licensing, etc. Getting the thing? Before marketing will come. That is the language of NCERT I wrote. So this is the first chapter over. Now we are going to deal about second chapter. So second chapter you keep in mind. Golden rice. Vitamin A rich rice. This golden rice is a GMO. Genetically modified organism or transgenic plant. Now flavor saver. It is also a GMO, genetically modified organism, and it is a variety of tomato. Here silencing of which gene occur? Polygalactourinase gene get silenced. So it is a polygalactourinase is a cell wall digesting enzyme. And how it silenced? Silenced by antisense technology. In antisense technology, we make the mRNA double stranded. So translation not occur. Able to understand. And so why this benefit of this G flavor saver? Increase the shelf life. Increase the shelf life of tomato. So this is about which one? Golden rice. Now we are going to talk about BT cotton. It is also a genetically modified organism. B stand for bacillus and T stand for thuringiensis. So in this cotton variety, gene for Bacillus thuringiensis is there. Actually what's happened? BT is a soil bacteria and these BT have cry-gene. Cry-gene synthesize a insecticidal protein. Insecticidal protein, and the name of protein is cry protein. Getting the idea? These cry protein is present in bacteria in inactive form. It present in bacteria as protoxin. So bacteria is not going to harmed by which one? These cry protein, and these bacteria release cry protein at certain stage of its life cycle. So through genetic engineering, what scientists have did, they remove the cry gene from bacteria and added it with a Agro Bacterium vector. Here it is which site? Here it is ori site. Ori site. And here it is what? They added the gene, cry gene with which one? Cry gene with one? With the vector DNA. With the vector DNA. Able to understand? So what happened? It become a recombinant plasmid. Now they transfer it into cotton cell. So cotton cell, who came here now? In cotton cell, who came here now? Here who came? Cry gene came along with a vector DNA which have ori site. Now through tissue culture, we develop a cotton plant. So this is cotton plant with every cell have cry gene. In every cell of the cotton, who is producing? Cry protein is producing. Now the caterpillar larval stage is going to feed on feed on plant part. Plant part will came into gastrointestinal tract and then it enter into the midgut. So in the gizzard, what happens? Churning of food occur, cry protein get released and they enter into the midgut. Midgut at alkaline pH. At alkaline pH, what happens? Cry protein, or you can say cry protein, inactive cry protein, or you can say protoxin converted into which one? Toxin. Converted into which one? Toxin. Able to understand? It becomes soluble. In inactive form, it was insoluble. In active form, it becomes soluble. And it happened at which pH? Alkaline pH. Getting the things. And these toxin, what they do, it bind with the epithelium. It bind with epithelium of midgut. And it create a pore. Through pore, water will enter, and after entry of water, lysis occur, insect is going to dead. So these are the way what happened. We make a crop resistance variety, which one? Insect resistance variety of the cotton. Cotton, who is the normal enemy? It is getting infected by bollworm. So bollworm resistance, it is an insect. So these bollworm resistance variety of the cotton we developed through this genetic engineering by introducing the cry gene in the cotton plant. So you also keep in your mind, these cry protein kills insect of how many order? NCERT wrote na, kills insect of order. You tell me which order is there? 3 order written in NCERT. What are the name of these 3 order which is given in NCERT? Tell me name of the 3 order. Diptera, Coleoptera and Lepidoptera. So it not kill all insect, it kill the insect belongs to how many? 3 orders. So I think it's required to take 10-minute breaks. Yeah? Is it okay? I also want some breathing space. Wait a minute. Yeah? Right. So 10 minutes of breathing space, and here it was somehow conceptual, so I was somehow slow. Now I will increase enormous speed, getting the things or not. Okay. So we will talk about that right now. So after RNAi, okay, RNAi, after 5 minutes we will take the break now. It is better. After finishing RNAi, it is okay. Okay, after RNAi, after 5 minutes, give a minute. RNAi. RNA interference, it is a method of cellular defense. By post-transcriptional gene silencing. By post-transcriptional gene silencing. During RNAi, another important line. During RNAi, the source of nematode specific gene is either RNA virus or transposome. It is a mobile genetic element. So we attach this nematode specific gene with vector DNA in such a way it produces double-stranded RNA. Another important line, vector we use during RNAi is which one? Agro Bacterium. And through RNAi, we make a variety of which one? Tobacco. NCERT wrote na AMI, Mylidogyne incognitia, infect root of tobacco plant. So we develop a such kind of variety. We develop a such kind of variety of tobacco. If MI is going to attack, MI is going to kill. So that much information about RNAi you have to keep in mind. Getting the idea. So after 10 minutes, I think 10 minutes or 15 minutes, what you want? 10 minutes? 15 minutes you want? 10, 15, different is coming. Okay, so 48, 58, I think 11 o'clock we will meet once. Okay, 11 o'clock we will start once. Okay. Recording start. Chat with host only. Recording start. So now we are going to deal about microbes. So I am telling about some product name. You have to tell me the bacteria. Citric acid, lactic acid, butyric acid. Getting the things. After that, I am telling you some product name. A statin, cyclosporine, a streptokinase, Swiss cheese, and Roquefort cheese. Tell me the microbes responsible for citric acid. Aspergillus niger, very good. Aspergillus. For lactic acid, Lactobacillus, lactic acid bacteria, lab. For butyric acid, hurry up. Clostridium butyricum, very good. Now, a statin produced by which one? A statin, Monascus purpureus. It is, these three are golden point. These three are golden point. A statin function of a statin. Blood cholesterol lowering agent. Cyclosporine A produced by cyclosporine A, Trichoderma polyesporum. What is this? Immunosuppressive agent. And a streptokinase produced by Streptococcus. And it is what basically? Clot buster. If we talk about Swiss cheese, produced by whom? Propionobacterium saramani. And Roquefort cheese produced by which one? Penicillium roqueforti. Penicillium roqueforti. So Roquefort cheese by fungi and Swiss cheese by bacteria. And you better know about which one? Baker and brewerist. Baker yeast and brewer yeast. Who is baker yeast and who is brewer yeast? Tell me. Saccharomyces cerevisiae. Cerevisiae. These are baker and brewing used in baking and brewing industry. Alcoholic drink without distillation. What are the name of alcoholic drink without distillation? Wine and beer. And rest of the alcoholic drink produced by distillation. So in wine and beer, alcohol content is what? Less. Because it produced without distillation. In whiskey, brandy, the alcohol content is high. It produced by process of which one? Distillations. Getting the idea or not? Now Now we are going to deal about microbes as biocontrol agent. Microbes as biocontrol agent. I am writing some name. Dragonfly and ladybird. Then I am writing trichoderma. Then I am writing first dragonfly and ladybird. Then I am writing trichoderma. Then BT. Then which one? Baculovirus. Tell me, dragonfly is a biocontrol agent against dragonfly and ladybird against? Who is against aphids and who is against mosquito? First tell me who is against aphid. Two different answer is coming. Mosquito, dragonfly. Ladybirds, getting the things? Aphids, very good. So, now trichoderma is a biocontrol agent against trichoderma, root-borne pathogen. And BT, Bacillus thuringiensis against insect. And baculovirus is a nucleopolyhedrovirus. And it is a narrow spectrum. It works against which one? Insect and some arthropods. So these are the biocontrol agents you keep in your mind. Next. Now, next I'm going to tell about sewage treatment. Two steps. Primary, secondary. Primary have two steps. Filtration and sedimentation. Primary is a physical process. Secondary is a biological process. It is a biological process. So if we talk about primary treatment, filtration and sedimentation, during sedimentation, two terms you keep in your mind. Primary sludge and effluent. Who is present in a primary sludge? It is what? It is in bottom, settled in bottom. Settled in bottom. And these primary sludge include small stone, sand, pebbles. Effluent, water plus dissolved organic matter is called effluent. Water plus dissolved organic matter is called effluent. If we talk, we perform secondary treatment of which one? Effluent. Effluent undergone in which treatment? Secondary treatment. So during secondary treatment, we are doing first step is aerobic. So we supply the air through air pump in a primary chamber, aerobic chamber, and here blades, rotator are there for mixing. So what's happened when we are treating the effluent, what happened? A lot of who will grow? Aerobic bacteria plus fungal hyphae, fungi. So these aerobic bacteria and fungal hyphae is called flocks. So these flocks is doing what? These flocks are digesting, or you can say decomposing, which one? Organic content. These are what basically heterotrophic bacteria, and we perform until and unless who is not going to decrease? BOD is not going to decrease. So we perform aerobic treatment, secondary treatment, first step until unless BOD is not decreased. BOD is very important term. More polluted water, more BOD. It is an indicator of water pollution. More BOD means more organic matter in the water. So these flocks going to reduce the BOD. After BOD is going to reduce, the flocks be transfer into which one? These are going to transfer into settling tanks. So these settling tanks, it is called activated sludge. Activated sludge. And effluent who are water with very less BOD be transfer in water body. We transfer in water body. And after that, these activated sludge going for which treatment? Anaerobic chamber. And in anaerobic chamber, what happened? It is going to produce which one? Biogas. Biogas. Biogas, a major constituent, which one? Methane. Getting the things or not? CO2 is also there. H2S is also there. Getting the things or not? And here which bacteria is there? Methanogen. So this is about the sewage treatment. I told even somehow step by step. Sewage treatment over. Biofertilizer. Biofertilizer. So if you talk about nitrogen fixing bacteria, like cyanobacteria, example, nostoc, Anavina, Oscillatoria, act as a biofactor, and these cyanobacteria present in paddy field. In paddy field, Azolla, Anavina association is there. Azolla is a fern, Anavina is a cyanobacteria. Another biofertilizer you can say about many microbes, nitrogen fixing microbes are there. Even free living nitrogen fixing microbes, many examples, free living and symbiotic nitrogen fixing microbe. Example, Azotobacter, Basiankia, Rhodospirillum. It is what? Free living, symbiotic, you can say Rhizobium, Frankia. These are symbiotic. Rhizobium with leguminous plant and Frankia with non-leguminous plant. So these are some of the nitrogen fixing microbes. If you talk about mycorrhiza, it is an association of glomus. It is a species of fungi with which one? Higher plant. An example, Pinus. So it is which one? Mycorrhizal association, where fungal partner absorb phosphorus provide resistivity against root-borne pathogen, help to tolerate with which one? Drought like condition. Able to understand? Mineral absorption. So these are some of the work performed by the fungal partner, and higher plant in return give them food. So it is a mycorrhizal association. Able to understand? So this chapter also over. Now we are going to deal crop improvement. All table of NCERT is important in crop improvement. Moong bean, some important things I am telling you. Moong bean produced by which one? Mutational breeding. Mutational breeding. And these moong bean is resistance to which one? Yellow mosaic virus. And another one, which one? Powdery mildew. Another things I am telling you. Prabharani Kranti. It is a breed of which one? And it is resistance against what? You tell me. Prabharani Kranti. It is breed of which one? Evelmascus esculentus, okra, bhindi, ladyfinger, and it is resistivity against which one? It is bhindi breed or okra breed, and it is resistance against yellow mosaic virus. How it develop? How it develop? Examiner will ask. Because both resistance to yellow mosaic virus, moong bean also, and this windy variety also, it by hybridization with whom? Here hybridization with wild relative. Hybridization with wild relative, wild variety. Getting the things or not? Now we are going to move ahead. Now we are going to tell you about SCP. Keep the variety in mind. Like IR8 is a variety of, Taichung native 1 is a variety of, Jaya is a variety of, Ratna is a variety of, Kalyan Sona is a variety of, Kalyan Sona is a variety of, Sonalika is a variety of, Sonalika is a variety of. So IR8 is a variety of. Up to here it is variety of which one? Rice. And these are variety of which one? Wheat. Atlas 66 is also a variety of which one? Wheat. Getting the things or not? So these are the variety of wheat. High yielding variety, semi-dwarf variety. Getting the idea? Understanding the idea? So these are semi-dwarf variety and high yielding variety. And semi-dwarf variety of rice made by Norman Borlaug by crossing between these two, IR8 and Taichung native 1. Able to understand? Getting the idea? International Rice Research Institute present in where? Philippines. And Indian Rice Research Institute present in. It is not in NCERT. Keep in mind Cuttack. It is in Odisha. A place named Cuttack. It is in Odisha. Now we are moving ahead on which one? Single cell protein. Few examples are what? Yeast is a source of single cell protein. Spirulina, these are a rich source of protein through a alternative agriculture. You can say about which one? Chlorella. These are some of the example of which one? Single cell protein. Another micro cyanobacteria, Methylophilus methylotropus. Methylophilus methylotropus. These are some of the single cell protein example. These are rich source of protein produced by unconventional agriculture. Now we are going to deal about tissue culture. Tissue culture. In tissue culture, first we are going to talk about what is tissue culture. So it is a technique. It is a technique. What is micropropagation? Producing thousands of plants through explant is called micropropagation. How we produce? By this tissue culture technique. Tissue culture, during tissue culture, what's happened? Vegetative propagation. It is a kind of asexual reproduction. In vitro, in lab. Getting the idea? If you talk about tissue culture, progeny are clone of parent. Progeny are clone of parent. What is totipotency? Ability of a single plant cell to produce whole plant is called totipotency. Explant. What is this? Part of plant used in tissue culture. Is called explant. So these explant we prefer meristematic tissue. Why? Because it is free from viruses. It is free from infections. Getting the things. So now we are going to deal another terminology of tissue culture, and that is which one? Callus. Undifferentiated mass of cell. Undifferentiated mass of cell is what basically callus. After callus, we are going to deal about plantlet. New plant formed by tissue culture. PG are used during tissue culture. Auxin, cytokinin. Auxin for which growth? Root growth. Cytokinin for which growth? Shoot growth. So auxin and cytokinins are used during which one? Tissue culture. Getting the idea? So this is about tissue culture. Getting the idea? Somatic hybridization. Somatic hybridization. How it will do? By protoplast fusion. We have taken a cell of potato, fused with the cell of tomato. So how we did it? First we use the enzyme callase, which one? Cellulase and pectinase. Cell wall will digest it. It is the protoplast. Then by electric spark, by using PEG, we fuse it. The cell which is produced is called somatic hybrid. So we develop tomato by this method. But what the disadvantage, what the bad luck we have? we not got all the desired character in the progeny of the pomato. So the method PEG, many times this asked in the exam, polyethylene glycol, it enhances the chances of protoplast fusion. Enhance the chances of protoplast fusion. So this is the chapter. Now we are going to deal about asexual reproduction. Not asexual reproduction, reproduction in organism. Reproduction in organism. What important to keep in your mind? Vegetative propagation. Who is going to use in vegetative propagation? So keep in mind all the underground and sub-aerial modification of a stem used in vegetative propagation. So you can say it is potato and here it is eye of potato. So it is a which one? Auxiliary word or lateral word. Used as a vegetative propagules. Rhizome, corn. Getting the things or not? These all are going to use in which one? Use as a vegetative propagules during vegetative propagations. Getting the idea? So I have written all the underground and sub-aerial modification of the stems used in which one? Vegetative propagations. And birds who are present in the underground or sub-aerial stem use as a vegetative propagules during the vegetative propagations. Getting the things? So if you talk about lifespan, not depend on size. Two similar size organisms may have different lifespan. So lifespan and life cycle, don't confuse. Two term is there. Period from birth to death is called what? Lifespan. Life cycle is what? Producing the progeny is what? Completion of life cycle. When an organism produce an organism like themselves, life cycle get complete, but lifespan not get complete. So lifespan and life cycle are two different terminologies. Getting the things or not? Now we are going to deal about steps during sexual reproduction. So what are the steps? Three phases are there. One is pre-fertilization event. Another is what? Fertilization event. And another is what? Post-fertilization occur. So pre-fertilization event is what? Gametogenesis. Fertilization, syngamy and triple fusion. And post-fertilization, embryogenesis. Fertilization is of two types, external, internal. When occur in female body, it called internal. And when fertilization occur in outside, in water, is called external fertilization. For external fertilization, synchronization in release of male and female gamete is important. They produce a large number of gamete, and the chances of the development of the organism is less. So in evolutionary process, what you find, gradually external fertilization taken over by which one? Internal fertilizations. Getting the idea? So now we are moving ahead on sexual reproduction. Sexual reproductions. So in sexual reproduction, if we talk about ovule, is also called which one? Megasporangium. Ovule is also called megasporangium. Getting the idea? So if you talk about, I'm just telling directly the fertilization part, because here you keep in mind, this is the stigma. Here pollen grain will landed either in 2-cell or 3-cell stage. After landing on the stigma, what you are finding, pollen tube is going to form during pollen pistil interaction, and these pollen, I'm going to tell you the double fertilization first. What happened? These pollen tube will germinate, and these germinating pollen tube moving through style providing the nourishment, and they are finally reaching to which one? Ovule, entering in the ovule. If it is, mostly it is entered through micropylarine inside the ovule, and inside the ovule, who is there? Embryo sac is there. In embryo sac, who is there? Egg apparatus is there. So what you find during double fertilization, egg fertilized with which one? One of the sperm nuclei or male gamete, a sperm nuclei or male gamete, and form what? Basically zygote. And when two polar nuclei fuse with a sperm nuclei, it is going to form a triploid structure, that is primary endosperm nuclei, and the event is called triple fusion. Double fertilization and triple fusion is the characteristic feature of which one? Angiosperm. Some outbreeding device name I am writing. Outbreeding device. Outbreeding device. What are outbreeding device? Unisexuality. Unisexuality is one of the outbreeding device. Dichogamy. Dichogamy is outbreeding device. It is which one? Protandry and protogyny. Both are outbreeding device. Getting the things or not? Pollen prepotency is a outbreeding device. It promotes cross-pollination, retards self-pollination. Getting the things or not? Another things you can say about the outbreeding device is hercogamy is a outbreeding device, and self-incompatibility is also a outbreeding device. These are the things who promote cross-pollination and retard self-pollinations. Getting the idea or not? Now I am going to tell you about which one? Apomixis. Apomixis is what? Production of seed without fertilization. So apomixis is a kind of asexual reproduction. It is a kind of asexual reproduction. If you find apomixis, it maintains the genetic constituent generation after generation. So nowadays, to avoid inbreeding depression, we are looking toward apomixis variety. If any seed who is a better variety of seed, what happen? It go for inbreeding depression due to sexual reproduction. If we develop by apomixis, its quality will remain same all the time. Polyembryony. Presence of more than one embryo. More than one embryo. Where we report polyembryony? In citrus. In mango, we report polyembryony conditions. Diplospory, apospory, apogamy not dealt in NCERT. I am not going to deal. So now we are moving ahead. Now we are going to deal about from where should I start? You tell me. From living world side I start or from plant physiology side you start? What you wish? Two areas there. Either start from plant physiology or living world side. Physiology side you want. Okay, no problem. So I am dealing about transport in plant. Transport in plant. So in transport in plant, what you keep in mind? Water movement or absorption ultimately occur due to suction pressure. Or DPD or water potential. Any language you can know. Suction pressure or DPD or water potential. So root is absorbing the water due to suction pressure. Water movement. Water movement direction. I am writing. Low to high DPD. High to low psi W. If DPD and psi W is not there, only one parameter is given. So you can say more psi S to less psi S. This is the direction of movement of water. More psi S to less psi S. Another direction of movement of water is less OP. If TP is not given, so we will deal in OP language now only. Less OP to more OP. If someone going to give you, this is a cell where another things is not given. OP equal to 5, here OP equal to 10. What will be the direction of movement of water? So A to B. Getting the idea or not? From hypotonic solution side to hypertonic solution side. Hypertonic solution side. So these are the direction of the movement of water from more free energy to less free energy. Many language I told. Free energy to less free energy. Water movement direction. You better know psi W equal to psi S plus psi P. If both parameter is there, we not answer water movement in any one parameter, we answer in which one? Psi W. Getting the things or not? Right? So water movement occur from these languages. Able to understand or not? Now we are moving ahead. I am moving ahead. If we talk about means of short distance transport. Distance transport. What are the means? Diffusion. Another means cytoplasmic streaming. And diffusion is of how many types? Two types. Simple diffusion and facilitated diffusion. and facilitated diffusion. These are the, another one is active transport. So examiner will ask, out of cytoplasmic streaming, diffusion and active transport, how many require ATP? So cytoplasmic streaming require ATP. Means it is a which kind of process? Active process. Diffusion not require ATP. Any kind of diffusion. Active transport require which one? At ATP. So who require ATP is a which kind of process? Uphill process. Getting the things? Both facilitated diffusion and active transport require a spatial membrane protein. Getting the things? Carrier protein. So who require for active transport is called pump protein. There is a spatial carrier protein that is called pump protein. And wherever protein is required, that is going for saturation. So simple diffusion, not require protein, not saturated. Because protein is not required. Facilitated diffusion undergo transport saturation, active transport undergoes transport saturation. Getting the idea or not? So these are about different type of diffusion. Now we are moving ahead. Now I am going to tell you about some correlations. Formula I am telling you. 1 minute. I already told psi W equal to psi S plus psi P. Side by side I am writing dPd equal to OP minus dP. What are the relation between those formula? Psi W equal to minus dPd, psi S equal to minus OP. Here it is solute potential, here it is osmotic pressure, and psi P equal to Tp. These are the relation between these two formula, and with the help of both formula, you can talk about water movement. What is the direction of the water movement? Getting the things or not? Understanding the idea. So these formula you can handle to use to handle what? Numerical based on what? Water movement. Able to understand? Getting the idea. I am telling you one thing also. Psi S is equal to Tp is equal to Wp. Wp is the wall pressure. Their value are always equal. Solute potential and osmotic pressure have same magnitude and opposite sign, and same with the water potential and dPd. Both have same magnitude with the opposite sign. Able to understand or not? Now, I am giving you some basic idea about apoplast and symplast. Apoplast and symplast. If we talk about apoplast, it is a passive process. Symplast usually active. Usually active. Depend upon. So apoplast is rapid. It is a which one? Slow process. Apoplast occur from where? It is non-living pathway. Apoplast is a non-living pathway. Non-living pathway means what? It occurs through non-living part. Symplast is what? Basically living pathway. It is a living pathway. So one is non-living pathway, another is living pathway. Getting the things or not? Living pathway means what? It occurs through symplast. What happened? Cell membrane involved. Cell membrane, it occurs through cell membrane, protoplasm, plasmodesmata. These all are going to involve in symplastic pathway. In apoplastic pathway, it occurs through cell wall and which one? Intercellular space. intercellular space. Getting the idea? So apoplast limitation is only one. What is the limitation of the apoplast? Apoplast limitations is apoplast pathway help only in transport up to endodermis. Apoplastic pathway not cross the endodermis. Casparian strips. Casparian strip, where it is present? Present on radial and tangential wall. Tangential wall prevent apoplastic pathway. So ultimately, in xylem vessel, water enter by symplast. Ultimately, movement of water and mineral in xylem vessel occur by symplast. Occur by symplast. Apoplast alone can't do it. Now we are moving ahead. Now I am going to talk transport of mineral. Keep in your mind. Transport of mineral. Loading, unloading, reloading. Keep in your mind. Loading from where to where? Root hair to xylem vessels. It is a which kind of process? Usually active. Loading is usually active. Without apoplast, it can't occur. Unloading always passive. And reloading of mineral is active. Unloading means what? You just try to understand through a diagram. Here I am drawing a diagram: epidermis, cortex, endodermis, pericycle, it is Casparian strips, and it is xylem vessels, and it is leaf. It is leaf. So what happened? Water and mineral reaching up to here by apoplast, it is apoplastic pathway, and water and mineral reaching up to here by symplast, or it may be from here it's symplast. So ultimately it is taking which one? Symplast. I am using another color, so you got the idea. This is symplast and this is symplast. So it is usually active. Symplast is mostly active. Now it is called loading. It is called loading. It is called loading. What is unloading? Unloading is from here to vein ending. It is unloading. And from vein ending to cell, inside a cell, from vein ending to inside a cell, it is called reloading. It is called reloading. So reloading is what? Vein ending to cell, it is active. Unloading, it is what? Passive. So these are about the how translocation of mineral occur. Getting the things or not? Mineral translocation, loading, unloading, and reloading, three things is there. Now I am going to tell you about ascent of sap. Ascent of sap. If you talk about ascent of sap by which one? Cohesive tension theory or transpiration pull theory. Most accepted theory for ascent of sap is this, given by Dixon and Joly. So what they told, due to transpirative loss of water, continuous suction pressure is going to create, and these continuous suction pressure pulling the water into the xylem vessel up. Getting the things? G is pulling another force. So another pressure, that is the root pressure, is just countering which one? Gravitational pull. Because gravitational pull pulling down, root pressure pushing up. So water moving, pull up, water pull down due to suction pressure. It is due to suction pressure. It is due to G. So water not going to break. Reason being is what? Water have high tensile strength. Water have high tensile strength due to cohesiveness, adhesiveness, and high surface tension. Any chances of breakage is there, that is overcome by root pressure. If any column break occur, root pressure going to push it. Now I am going to tell you about root pressure and guttation. Root pressure and guttation. Root pressure and guttation. Root pressure is a positive hydrostatic pressure. Positive hydrostatic pressure. Second thing, pushing pressure. It pushes the water into xylem vessels. Third things, it is due to active absorption of mineral. So it gives a small distance push of water. Push of water in xylem vessels. Getting the idea? Guttation is what? Guttation, loss of water in impure liquid droplet from hydathode. Hydathode is what? Water stomata. Where it is present? Leaf apex. Present at leaf apex. Getting the things or not? Present at leaf apex. Able to understand? It's called guttation. So loss of water in impure liquid droplet from hydathode, that is water stomata, present at leaf apex, is called guttation. Guttation occur in which one? Herbaceous plant. Occur in, oh, I'm writing in another slide. Guttation occur in a small herbaceous plant. A small herbaceous plant. It is due to high root pressure. Why guttation occur? High root pressure. Which time guttation occur? It occur in early morning or midnight. Midnight of warm, humid summer. Warm, humid summer. When guttation occur when humidity is high and transpiration is low. Temperature is high, transpiration is low. Humidity is high, obviously, so root pressure will more, transpiration is less, so that time guttation occur. Now I am going to tell about translocation of food. I already told about the stomata, not much mechanism is given. So translocation of food. If we talk about translocation of food, occur through phloem. occur through phloem. It is what? Bidirectional process. Bidirectional process. Getting the things? Girdling experiment proved translocation occur by which one? Phloem. In girdling, what we remove? Remove the bark, removal of bark. So when we remove the bark, food translocation not occur. No food translocation. You keep in mind. No food translocation will occur when we remove which part? Bark. Getting the things. Understanding the idea. So if we talk about two things is there, which you will keep in mind, it occur by mass flow. Either any long distance transport occur by mass flow, either through xylem or phloem. Mass flow. This mass flow is due to difference in pressure of two sides. Difference in pressure of two sides is what basically mass flow. During translocation of food, food move from where to where? High turgor pressure to low turgor pressure. Through phloem it occurs. So difference in pressure is there now. One side high TP, another side low TP. So mass flow occur. Mass flow is what? Movement in mass. Solute and solvent move together. Able to understand or not? So two important steps is there: loading and unloading. Here reloading term is not there. Reloading term is in mineral. So loading is what? From mesophyll cell to, when food move from mesophyll cell to sieve tube, it is called loading. It is an active process. And when food move from sieve tube to sink cell, it is called unloading. Unloading is also active process. So both loading and unloading during food translocation is an active process, require ATP, and translocation of food occur from high turgor pressure to low turgor pressure. You got the idea? It occurs from high turgor pressure to low turgor pressure. Transpiration is directly proportional to temperature. Transpirational is inversely proportional to humidity. Transpiration is inversely proportional to CO2 concentration. If CO2 concentration will increase, transpiration will. Transpiration is depend mainly on which one? Vapor pressure. It is directly proportional to difference of vapor pressure of two sides. Getting the idea? So transpiration, as much difference in the vapor pressure of the two sides, what happened? Transpiration is maximum. Suppose that there is a leaf and vapor pressure is 100%. Beside of that, who will open? A stomata will open, but who will not come out? Water vapor will not come out. Transpiration will not occur. Suppose that here air is dry, 0% water vapor is in the atmosphere, maximum transpiration will occur. Able to understand? So this is about the transpiration. Transpiration, you know better. Loss of water from exposed surface of plant is called transpiration. Now we are going to deal about photosynthesis. Photosynthesis. So if we talk about photosynthesis, first I am telling you the dark reaction side, C3 cycle. So in C3 cycle, rather than to write making cycle is more easy, I can make the cycle in one minute. So give your attention. So if we talk about C3 cycle, it is given, it is also called Kelvin cycle because this cycle is given by Melvin cycle. It is the ultimate pathway of glucose synthesis. Without C3 cycle, in any type of plant, glucose synthesis not occur. So during C3 cycle, what you are finding, one 5-carbon compound react with CO2, undergoing carboxylation in presence of enzyme Rubisco. Able to understand? So carboxylation occur, Rubisco act as carboxylase here, and formation of a 6-carbon intermediate occur, which break down into 2 3-carbon intermediate, that is the PGA, is the first stable product. Is the first stable product. Where this C3 cycle occur? This C3 cycle in C3 plant. In C3 plant occur in which one? Occur in mesophyll cell. Occur in mesophyll cell. First stable product is 3-carbon compound phosphoglyceric acid. So it is called 3-carbon compound. So it is called C3 cycle. Now the second step is reduction. And at reduction step, what happened? PGA, 2-PGA convert into 2-triose phosphate and phosphoglyceraldehyde. So here for each 1 ATP required, 1 NADPH required. So before reduction, what happened? Phosphorylation. So PGA undergoes in phosphorylation and that require ATP, then goes in reduction which require NADPH and converted into triose phosphate. Here as a byproduct who release inorganic phosphate. Now this triose phosphate undergoes in third step, that is the regeneration, which require ATP converted into ADP and RUBP is going to generate. So one CO2 fixation require 1 Kelvin cycle. So during glucose synthesis, 6 CO2 fixation occur. So how many cycle will require? 6 Kelvin cycle required. So for 6 Kelvin cycle, you multiply everywhere with what? 6. Multiply everywhere with what? 6. Getting the thing? So here you got 12 PGA, here you got 12 triose phosphate, here you require what? 12 ATP, 12 NADP, 12 inorganic phosphate required. And out of 12, 2 get exported. 2 export, 2 3-carbon compound get export and form a 6-carbon compound glucose. So out of 10, 12 to export, 10 use in regeneration and regenerating how many molecule? Regenerating 6 molecule of RUBP. 6 5 is 30 and 10 3 is 30. Triose phosphate is a 3-carbon compound. So during C3 cycle, for each Kelvin cycle, it requires how many? 3 ATP and 2 NADPH. 2 ATP required at reduction step, 1 ATP required at regeneration step, and only 1 ATP required at which stage? Sorry, 2 ATP required at which step? Reduction. So at reduction step, 2 ATP and 2 NADPH require. 1 more ATP required in which one? Regeneration step. So total for glucose formation, 6 cycle required. So you multiply by 6, 18 ATP required and 12 NADP required in the C3 cycle. C3 cycle over. Now I am telling about C4 cycle. I am telling about C4 cycle. So in C4 cycle, what present? C4 cycle occur in C4 plant. Occur in C4 plant. In C4 plant, what you are finding? Carange anatomy is present. Carange anatomy is present. So if you talk about carange anatomy, what's happened? 2 type of cells are there. 2 type of cells are there. One cell is what? Mesophyll cell is there. Mesophyll cell is there. In the leaf, another who is present, bundle sheath cell is there. So if you talk about mesophyll cell, what you will find here? Chloroplast have grana. Here chloroplast have grana. Getting the idea? Here in bundle sheath cell, chloroplast do not have grana. It only have a stroma. Getting the idea? Here in mesophyll cell, who is absent? Rubisco is absent. So here dimorphic chloroplast is there. Here in bundle sheath cell, Rubisco is present. So Rubisco require for dark reaction and Grana require for light reaction. So in mesophyll cell of the C4 plant, who occur? Light reaction occur, and who not occur? Dark reaction not occur. Dark reaction not occur. Because who is absent? Rubisco is absent. In bundle sheath cell, only what occur? Dark reaction occur. Only dark reaction occur. So how you beautifully current anatomy help to avoid photorespiration? You got the idea or not? C4 cycle why it occur? To avoid photorespiration. To avoid photorespiration. Getting the thing? C4 cycle occur to avoid photorespiration. Getting the idea? So what you find, children, you just try to understand. Here light reaction occur, oxygen evolved during light reaction. Who is not there? Rubisco. Rubisco is in this cell. So these oxygen can't enter into bundle sheath cell because bundle sheath cell are compactly arranged and they have an impervious wall for the gaseous exchange. So these oxygen can't enter into the cell where Rubisco is there, and these oxygen move into the environment. So Rubisco not pressurize the bundles, pressurized by oxygen to perform photorespiration in a C4 plant. Now question arises, how photosynthesis occur in a C4 plant? So photosynthesis occur, you just have a look. How CO2 fixation occur? So this is two type of cell. Mesophyll cell, here it is bundle sheath cell. You please try to understand. Here who is coming? CO2 is coming, reacting with PEP, 3 carbon compound, converting into OA, 4 carbon compound. Able to understand? First CO2 react with water, but I am not writing that all steps. So oxaloacetic acid is the first stable product. And it form in which one? Mesophyll cell. It first CO2 fixation occur in mesophyll cell. Now oxaloacetic acid convert into which one? Malic acid. And these malic acid form by the process of reduction. So it require NADP, NADPH. So NADP. Able to understand? Now malic acid through what? Pit. Enter through plasmodesmatase. Pits enter into which one? Bundle sheath cell. Now this malic acid in bundle sheath cell converted into pyruvic acid. Here oxidative decarboxylation occur, CO2 release and hydrogen release in the process of oxidation. So what you are finding, these CO2 who release in bundle sheath cell is enter into which cycle? C3 cycle. So without C3 cycle, also in C4 plant, photosynthesis not occur. Here first stable product is PGA. So examiner will ask, PGA is the first stable product of carboxylation. Second carboxylation, PGA is the product formed by second carboxylation. Oxaloacetic acid is the product formed by first carboxylation. In C4 plant, first carboxylation occur in mesophyll cell, second carboxylation occur in bundle sheath cell. So what require in C3 cycle? It will require here or not? 3 ATP required, 2 ADP required. Now this pyruvic acid enter into which cell? Mesophyll cell, and it converted into PEP. In this process, ATP converted into AMP, which is equivalent to 2 ATP. So for one CO2 fixation, in C4 plant, how many? 5 ATP. What is the breakage of 5 ATP? 3 in C3 cycle and 2 ATP in regeneration. Two ATP in which one? Regeneration required. 5 ATP bifurcation is this: 3 in C3 cycle and 2 in regeneration ATP required. And how many? And 2 NADPH required. NADPH same like C3 cycle, ATP is more. So additional 2 ATP required in C4 plant for every fixation of which one? For every fixation of which one? 1 CO2. So for 6 CO2 fixation, how many ATP required? 30 ATP required in which one? C4 plant. Able to understand or not? Now we are moving ahead toward which one? Photorespiration. Photorespiration or C2 cycle. Or C2 cycle. If we talk about photorespiration, or C2 cycle, what happened? RUBP react with O2. So when RUBP react with O2, in presence of which enzyme? Rubisco enzyme. Rubisco here work as oxygenase. It break into two type of product. One is forming PGA. One PGA is forming and one is forming 2-phosphoglycolate. So these PGA enter in which cycle? C3 cycle, and these 2-phosphoglycolate is first stable product. And it is a 2-carbon compound, and that is the reason it is called C2 cycle. So during photorespiration, what's happened? No photosynthesis. No photosynthesis. This photorespiration is what? Photooxidation. Occur only in light. Only in light. Able to understand? Getting the idea? So it is different from respiration. So here photosynthesis not occur, photooxidative damage occur, and that is the reason many times people throw it is a wasteful process. But still we don't know the physiological function of the photorespiration in the plant, why this photorespiration is going to occur. So this is about the C2 cycle. Now I am going to tell you about chemiosmotic and light reactions. Chemiosmotic hypothesis. So give your attention. Here it is PS2. Here it is B6F. Here this is PS1. So what's happened during electron transport, non-cyclic or cyclic mainly non-cyclic photophosphorylation, electron move from PS2 to BS6 and from PS1. PS2 to cytochrome B6F, and then electron move to which one? PS1. Getting the idea? So when these electrons are moving, what you are finding? Energy is releasing. When electrons are moving, what you are finding? Energy is releasing. And when these energy is releasing, what happening? Proton is pumped from where to where? A stroma to lumen. Initially, both side equal proton was there. When light reaction occur, electron of PS2 settled in which one? PS1. And during this movement, electron move downhill in potential energy scale. When electron move downhill, energy will release. What is the use of this energy? It is pumping the proton from stroma to lumen. When from PS1, electron is shifted into NADP plus. So what's happened? Proton have taken from stroma and it is converted into NADP. NADP plus is converted into NADPH. NADPH. Able to understand or not? Getting the idea? So here loss of proton to whom? Stroma. And so what you are finding during non-cyclic photophosphorylation? PS2 electron settled in PS1. PS1 electron settled in NADP. And what happened? Ultimately, who is in the loss? PS2 is in the loss. It acts as a potent oxidizing agent. So in inner side of the membrane, thylakoid membrane, getting the things or not, or frit membrane, OEC is there. That OEC is going to photolysis of water. Photolysis of water, examiner will ask where it occur. It occur in inner side of the membrane. It convert, it break into 2H plus half O2, from 2H, electron will move to PS2, proton will release into lumen. So what you find? Earlier, both side equal proton was there. Due to light reaction, proton came from outside to inside. Loss of proton occur into the stroma. Income of proton occur into the lumen. And due to all these reasons, proton concentration increase into which one? Proton concentration increase into inside. Able to understand? The lumen. And when these proton move out, getting the idea or not? When these protons move out, what happens? ATP produce. ADP plus inorganic phosphate attach and ATP produce. These protons move out through which one? CF node and CF1 particle. Here ATP synthetase enzyme is there. Getting the idea? So it moves out by which process? Facilitated diffusion. And it enter into which by process? Active transport. So ATP production occur in such a way by which one? Chemi-osmotic hypothesis. Now we are going to deal about which one? Factors affecting photosynthesis. Factors affecting photosynthesis. Hanji. Factor affecting CO2 and light. I am writing two things here only. So CO2, C3 plant, C4 plant. I am writing saturation point. CO2 saturation point is 450 ppm in C3 plant, and in C4 plant it is which one? 360 ppm. Light as a factor, if you talk, initially when the light intensity increase, photosynthesis increase, but after a level it stop because light not remain a limiting factor. So what is law of limiting factor? Examiner will ask, law of limiting factor given by Blackman. And what he told in this factor? He told at a given time, the factor who is present in minimum amount will regulate the process. So, getting the idea, these are the factors. Temperature as a factor affect which one? Affect dark reaction more. Dark reaction more because it is a enzymatic process. Now we are moving ahead. Now we are moving ahead on respiration. Respiration. Another 10 minutes. I want to finish physiology, then I will move another topic. So respiration. So if we talk about respiration, see glucose converting into pyruvic acid. 1 glucose into 2 pyruvic acid in the process of glycolysis. Glycolysis require ATP at 2 different steps. During glycolysis, at one step, production of 2 NADH occur, not NADPH. And at 2 different steps, production of 2 ATP occur. And these ATP production occur due to substrate level phosphorylation, SLP. So these happen during glycolysis. After pyruvic acid, these pyruvic acid during aerobic respiration undergoes in which one? Link reaction. Link reaction and undergoes in link reaction, and during link reaction 2 pyruvic acid converted into 2 acetyl coenzyme A. During that process, what you are finding? 2 molecule of NAD plus converted into 2 molecule of NADH. Able to understand? And decarboxylation occur, 2 CO2 also released from 2 pyruvic acid. Each pyruvic acid 1 CO2. Some cofactor require magnesium ion, coenzyme A, getting the things or not, and acetyl coenzyme A is forming. These link reaction occur in matrix of mitochondria, and it occur in presence of oxygen. Glycolysis occur in cytoplasm, and these glycolysis occur in both presence or absence of oxygen. Means if oxygen is there or not there, no impact on the glycolysis. Glycolysis will occur. Now these acetyl coenzyme A undergoes in which one? Krebs cycle. And when Krebs cycle occur, what you are finding? Oxaloacetic acid. I am talking about one Krebs cycle. You multiply with 2, all result will come. Oxaloacetic acid react with one acetyl coenzyme A. I am taking only one, and it is going to produce which one? It is going to produce citric acid. Citric acid is the first stable product. So that is the reason this Krebs cycle is also called citric acid cycle, and this is a tricarboxylic acid, and that is the reason it is also called TCA cycle. And then it is going to convert into what? You better know, alpha ketoglutaric acid. So during this step, conversion of citric acid into alpha ketoglutaric acid, what you are finding? NAD plus converted into NADH, oxidation occur, and also decarboxylation occur. Now alpha ketoglutaric acid converting into succinic acid, and during these conversion, succinic acid, and during these conversion also a CO2 is releasing, and you are finding NAD plus converted into NADH. Able to understand? Now succinic acid converted into which one? Malic acid. Malic acid. Getting the things or not? Now malic acid, when it is going to forming, during this process you also mind it, ATP or GTP produce. ATP or GTP produce, any of this. And when this malic acid is forming, what is producing? FADH2 is producing. And when this malic acid converting into oxaloacetic acid, NADH is producing. So from one Krebs cycle you find this. So how many total Krebs cycle will operate? 2 Krebs cycle, because 2 acetyl coenzyme is there. So everywhere you can multiply by how many? 2. So your result will come. Everywhere you can multiply by how many? 2, 2, 2. So what you are finding? This is about the Krebs cycle. So what we are finding on the overall energy, balance it. If we talk about glycolysis, what are the input? Energy require, energy release. Energy require, energy release. So glycolysis, what require? 2 ATP require, and 4 ATP release by SLP, and what you are finding? 2 NADH2 produce. NADH produce. Now I am going to tell you about which one? Glycolysis over. I am going to tell you about link reaction. During link reaction, no energy required, no ATP required, but who produce? 2 NADH produce. Now I am going to tell you about 2 link reaction. 2 Krebs cycle. So from 2 Krebs cycle, ATP required is 0. ATP produce is 2 during SLP from 2 glycolysis. FADH2 produces 2 FADH produce. Getting the things or not? And what you can say, NADH produces how much? 6 NADH produce. NADH produce. So these are the output of Krebs cycle. So 6 NADH guna multiply 3 equal to 18 ATP. Getting the things or not? 6 NADH equal to 18 ATP, 2 FADH here multiply by 2 equal to 4 ATP. So 18 or 4, how much? 22, and 2 direct ATP through SLP equal to 24 ATP. So 24 ATP came by 2 Krebs cycle. Each Krebs cycle give how much? 12 ATP. 2 link reaction given 2 into 3 equal to 6 ATP. Getting the idea or not? Now I am going to tell you about here in 2 NADH give 2 into 3 equal to 6 ATP. So 6 ATP, 4 produce, 6 plus 4, 10 ATP, 10 minus 2 equal to 8 ATP. Able to understand or not? So these are the balance sheet. So how many ATP produced by SLP? Total NADH is 6 plus 2 plus 2, 8 plus 2, 10 NADH. So 10 NADH producing 30 ATP, 2 FADH producing 4 ATP, 34 ATP come by which process? 34 ATP come by which process? Oxidative phosphorylation. And 4 ATP produced by which process? Substrate level phosphorylation. That is the total 38 ATP produced by complete oxidations of the glucose. Getting the idea or not? Understanding the idea. Now if we talk about ETS, it is a damn easy. ETS, it is a damn easy. Give your attention. Here examiner will ask what? They will ask the name of complex. Complex 1, complex 2, complex 3, complex 4, complex 5. So here 1, 3, 2, 4, 5 complex. Here what enzyme is there, you keep in mind. NADP dehydrogenase, FADH dehydrogenase, cytochrome BC1 complex. Here cytochrome AA3 and 2 copper center. Here ATP synthetase. Here mobile carrier is there. Which one? Coenzyme Q. Able to understand? Here who is there? Cytochrome C is a mobile carrier present toward perimitochondrial space. Now what happened in ETS? When NADH2, NADH plus H plus undergoes in oxidation and converted into NAD plus, electron moving by this pathway. Final acceptor of electron is half O2. So when electron from NADH moving through various carrier, energy release, and these energy used in pumping of proton. So 4 proton pumped at which level? Complex 1 level. Same 4 proton pumping occur at complex which one? 3 level, and 2 proton get pumped at which one? 4th level. So what happened? 4 plus 4 plus 8 total 10 proton get pumped whenever NADH is going for oxidation, and electron finally received by half O2. And here what you are finding, 2 proton it take from matrix and converted into water. So during each ETS half O2 require, 1 water produce. Final acceptor of electron, and see examiner will ask, final acceptor of electron is what? Half O2. Oxygen atom. 1 ETS, 1 half O2. So total how many ETS will require for complete oxidation of glucose? 12 ETS. How 12 ETS require? How 12 ETS require? Tell me, how 12 ETS require? Because 10 NADH2, 2 FADH is there, so 12 ETS require. Getting the idea? Understanding the idea? Is it clear in your mind? Okay. Right? So this is about ETS. 10 ETS when return back to maintain the equilibrium, how many 3 ATP going to produce? When 10 proton returning back, it is giving how many? 3 ATP, and these ATP produced by which one? Oxidative phosphorylation. Respiration is a amphibolic pathway. Energy not came only from which one? RQ I am going to tell you. Not coming by glucose only, fat and protein giving also. So carbohydrate RQ equal to 1. Protein RQ equal to 0.9. Lipid RQ is around 0.7. Organic acid RQ is, you will find greater than 1. Anaerobic respiration RQ is either infinite or not defined. RQ of the fatty seed is less than 1. So these are the respiratory quiescent and end of the chapter. Now I am going to tell you about mineral nutrition. Keep the name of bacteria in mind. So mineral nutrition. Mineral nutrition. Macro, micro nutrient you keep in mind. Macro is what? Carbon, hydrogen, oxygen, nitrogen, sulfur, magnesium, phosphorus, potassium, calcium. These are macronutrient. Micronutrient who is there? Iron, manganese, copper, molybdenum, boron, nickel, chlorine and zinc. These are 9, these are 8. These macronutrients require in what? Greater than 10 millimole per kg of dry weight. It require in what? Less than 10 millimole per kg of dry weight. Getting the things. So these are about the macro and micro. These are what? Basically essential element. Essential element are what element? Who have direct role of metabolism, who do not any substitute, and who require for completion of the life cycle of plant. So these total 9 plus 8 equal to 17 essential element is present in the plant. Getting the things. Now I am telling you about manganese toxicity. Table you mug up. Boron function, that table is very important. So manganese toxicity, what is the symptom? Symptom is brown spot with chlorotic vein. Brown spot with chlorotic vein. During manganese toxicity, what's happened? Deficiency of iron and magnesium occur. And also, what happened? How it occur? Because manganese perform competitive inhibition during its absorption. During its absorptions. So, another what happened? Calcium deficiency occur. How calcium deficiency occur? Manganese interfere with carbohydrate calcium translocation. It interfere with calcium translocation. So these are about the manganese toxicity you can keep in mind. Now I am going to tell you about nitrogen cycle. Nitrogen cycle have 4 steps. First step, nitrogen fixation. It is called disotropy. When it performs biological nitrogen fixation. Atmospheric nitrogen converted into which one? Ammonia. Getting the things. And during these atmospheric nitrogen fixation, I am writing a reaction here only. I am writing a reaction here only. Getting the things or not? What happened? N2 require 8 electron plus 8 proton plus 16 ATP converting into 2 ammonia. Here which enzyme require? Nitrogenase enzyme require. These nitrogenase enzyme work in oxygen free environment, and who is going to produce? H2 is going to produce. Able to understand or not? So for 2 ammonia, 16 ATP required. For each ammonia, how much? 8 ATP required. So these nitrogen fixation require nitrogenase enzyme, and these nitrogenase enzyme present in few prokaryote. Present in few prokaryote. It present in few prokaryotes. Example, rhizobium. Example, nostoc. Example, enabina. Example, Bajeringia. Example, rhodospirillum. A lot of nitrogen fixation bacteria is there. Getting the things or not? Now after that, what happened? I'm going to deal about nitrification. During nitrification, ammonia first convert into nitrite, and nitrite first then convert into nitrate. And these ammonia to nitrite is performed by nitrosomonas and nitrococcus. And nitrite to nitrate convert by nitrobacter. Getting the thing, these all three are chemosynthetic bacteria. Chemosynthetic bacteria. Plant absorb nitrogen mainly as nitrate. Plant absorb nitrogen mainly as nitrate. Mainly as nitrate. Getting the idea, understanding the idea. So this is about what basically nitrification. Then I am telling you ammonification. What happened during ammonification? You try to understand. During ammonification, dead and decaying organic matter, dead and decaying organic matter release which one? Ammonia. An ammonifying bacteria is Bacillus ramosus, Bacillus mycoides, Bacillus vulgaris. These are some of the bacteria who perform the ammonification. After ammonification, I am going to deal about denitrification. Denitrification. Denitrification is what? Nitrate, nitrite, or ammonia going to convert into atmospheric nitrogen, and it is done by whom? It is done by Pseudomonas and Thiobacillus. Getting the things. So this is about denitrification. Reductive amination. What happened during this time? Ammonium ion react with alpha ketoglutaric acid. Reduction require NADP. Glutamic dehydrogenase is the enzyme converted into glutamic acid. Transamination, conversion of one amino acid to keto, keto to amino. Amination. An example I am telling you, pyruvic acid react with glutamic acid, converting into alanine and alpha keto. So glutamic acid is a amino acid. Pyruvic acid is a keto acid. Pyruvic acid convert into alanine. So keto convert into amino acid, and glutamic acid convert into alpha keto. So amino acid converted into keto acid. So this is what basically, this is transamination. So mineral nutrition over. Now we are going to tell about, we are running for 15 minutes. Yeah. So plant growth. If I talk about plant growth, PGR, I will talk auxine. Auxine causes apical dominance. Another functions of auxine, if you talk about, delay senescence in young leaf and fruit, and promote in old. Promote in old region. Another functions of the auxine, you can say parthenocarpine tomato, in tomato. Another functions you will find about the auxine is what? Act as weedicide Example 2,4-D. Another functions of the auxins you can tell. So getting the things or not? Fully seedling disease caused by which one? Gibberellic acid, na? So another functions of the auxin is which one? Xylem differentiation, rooting at stem cut, getting the things. So this is about the auxin. Gibberellic acid main function. Gibberellic acid growth promoter, break dormancy. How can break dormancy? In seed, it going for conversion of which one? A stored food into the nutrient remobilization. It converts starch into maltose, maltose into glucose. Glucose will burn, energy will give. So that kind of work done by which one? Gibberellic acid. Break seed dormancy. Another internode elongation. Internode elongation. And if you talk about gibberellic acid, increase the productivity of the sugar cane, bolting. These are the some of the, it delay senescence. All growth promoter delay the senescence. So these are the some of the main function associated with which one? Gibberellic acid. Now we are going to deal about cytokinin. If we talk about cytokinin, it is also a growth promoter. This cytokinin promotes cytokinesis. If we talk about cytokinin and other functions, what you can say about the cytokinin and other functions, right? You can say chloroplast and new leaf formation. Getting the things. It is responsible for lateral branching. In that way, it is opposite to, in this function, it is opposite to which one? Auxin. That causes apical dominance, it causes which one? Lateral branching. It is also delay senescence by nutrient mobilization. So these are some of the function of cytokinin. Now we are going to deal about ABA. It is a inhibitor, growth inhibitor. Inhibitor do what? Promote aging and senescence. Promote abscission and senescence. It causes seed dormancy. Seed dormancy is caused by which one? ABA. Seed growth occur due to whose activity? Seed growth occur due to abscisic acid activity. And in many function, it is what? Antagonist of GA. I said many things in single word. Now I am going to talk about ethylene. It acts as both promoter and inhibitor. But mainly inhibitor. Promoter and inhibitor. Getting the things, it is a gaseous PGR. Ripening of fruit. Who is responsible? Ethylene. Marketed as what? Ethophone. It is responsible for what? A stem elongation in rice seedling. It also have a role in root hair formation. It also causing triple response. Thinning of cotton, walnut. These are the work done by whom? Ethylene, which is a gaseous PGR. So this is about the PGR. Now 2 lines about vernalization. Photoperiodism, nothing given in NCERT, you know, that is the reason I am not talking. Vernalization I am writing. So if we talk about vernalization, what is this? Vernalization means cold treatment. Vernalization means cold treatment. So it prevent precocious reproductive development. Precocious reproductive development. Late in growing season. So vernalization is a cold treatment required for before flowering in the plant in some of the variety. So in the variety, wherever vernalization require, it not flower until unless it not exposed with which one cold treatment. So natural vernalization give more time for vegetative growth. So it is a method by which nature give plant more time for vegetative growth. So a healthy plant is going to grow. Getting the things. NCERT wrote 2 type of varieties there. Of which one? Wheat and rye. So winter variety and spring variety. A spring variety do not require vernalization. Winter variety require vernalization. If we sow the winter variety in a spring season, it become biennial. So better we sow in which season? Autumn season. So in same season it is going to grow. Now I am going to tell you about which one? Cell. Cell organelle and function I am writing. I am starting with which one? Golgi complex. Here it is going to ask Golgi complex. Important functions of Golgi complex is what? Glycosylation. That is formation of glycoprotein and glycolipid. Glycosylation. Another functions is what? If you talk about cell secretion. Cell secretion. Another function is what? Packaging. So Golgi complex is a polar cell organelle. It have 2 phases. One is cis phase and another is trans phase. Cis phase is toward nucleus, trans phase is toward plasma membrane. Cis phase is called forming phase and trans is called maturing phase. So endoplasmic reticulum vesicle carry the protein or lipid fuse with the cis phase. Both are interconnected, cis and trans. It moving toward trans phase. During that time, what's happened? Chemical modification occur in Golgi complex. And after chemical modification, these chemicals are packed in Golginian vesicle. And these Golginian vesicle transported to different target. Many times what's happened? Many Golginian vesicle fuse together. And when they fuse together, they are forming a new cell organelle. That is what basically lysosome. So Golgi body is a cell organelle who is responsible for formation of lysosome. Golgi body is also associated with cell wall synthesis, cell wall formation. Cell wall formation. So these are some of the important functions of Golgi complex. Now I am going to tell you about ER. ER, endoplasmic reticulum. If we talk about endoplasmic reticulum, what you are finding? It is responsible for intracellular transport. Intracellular transport. It divides the cytoplasm into 2 components. So if we talk about RER and ACR, 2 type of ribosome. Which one is there? ER is there. RER have ribosome. ACR don't have ribosome. Don't have ribosome. If we talk about RER associated with protein synthesis, it is associated with lipid synthesis, lipid or steroidal hormone synthesis. It is done by which one? ACR. ACR also perform detoxification of drug. detoxification of drug. RER is a endoplasmic reticulum is a cell organelle. It performs protein folding. Protein folding means primary to secondary, secondary to tertiary structure form in which one? Endoplasmic reticulum. Endoplasmic reticulum form Golgi body. So Golgi body form from endoplasmic reticulum. And lysosome form from which one? Golgi body. So how many endomembranous system, how many cell organelle contain? ER, Golgi body, lysosome and vacuole are forming the endomembranous system. Why they are forming endomembranous system? They have a coordinated function. Coordinated function. Getting the idea or not? They have a coordinated function, so they are acting as a which one? Endomembranous system. Getting the idea? Now we are moving ahead. Wait, what's happening? Oh, slide over. So it's not moving now. Let me take a screenshot, otherwise it will not go. It become 99 slide, it is not moving on 100. Just a minute. You take a screenshot for this and just a minute. Let me take on the WhatsApp as well. After that, I will start afresh. Just a minute. 99 slide. I think it is not shifting on 100. So share. Wait a minute. It will take some time, 1 or 2 minutes. Have patience. Have patience. I will send to you, na? Thank you, dear. Mitochondria DNA is double-stranded, circular, single. Single, circular, and double-stranded. If you will say, I want to take 1 hour more, so I will finish plant. What? I think your lunch break, na? I got only 4 hours. I just come with a mind of 5 hours. Right? To finish also what? Plant kingdom, living world, morphology, anatomy, and biomolecules. That is remaining, na? Cell cycle also. So one day I will take 1 hour for all. I think now it is not possible today. So not possible today. I think it is. So any other day, right, na? So even your time of test is there, na? Food time and all. So you can go, I will take any other day. Right, na? Yeah. Thank you, children. Thank you very much for patience listening. Now you're feeling somehow comfortable, continuous class. Thank you. I think it came also. Yeah, bye. It's all become due to your cooperation. Thank you very much. Thank you, children. Thank you. Yeah, bye. See you soon. Yeah. Wait a minute. Recording is there. Recording I'll go see. Yeah. Save it. Save the recording. It is very important to save.